It's later. Sorry. Okay. So if we start with a situation like this and shrink the habitat size, eventually we are going to see a shift in the peak of the population to the left of the attractive location, okay? Because if they are to the right of the attractive location, they will die more commonly here on the right matrix, okay? So it is interesting to note that individuals may keep their movement patterns even if you change the landscape, okay? This is something that has been observed for some species, and it's called historical geographical range, okay? Eventually, there might be a situation where the attractive location is outside the habitat, okay? So the watering hole or the attractive part, it could be a chemoattractant. It is located outside the habitat. Even in this case, the population may still survive, okay? This will occur if the population can move around and interact and reproduce inside the habitat before going to the bad part, okay? So this indicates that there is a window of opportunity for conservation biology, okay? So eventually, if we keep decreasing the habitat size, we are going to see a critical size. Without attractive locations, the critical habitat size is proportional to the diffusion rate. So individuals that move more, that spread more, they need more habitats in order to avoid extinction, okay? Because they are spending more time in the matrix, in the bad part. However, if we consider attractive locations, this is not necessarily true. And the reason is essentially that if the attractive location is close to an edge of the habitat, then you are better off not going straight to that part. So if you diffuse more, instead of going straight to the attractive location, which is near a bad part, this is going to be good for you. So in this particular scenario, more diffusive individuals may require less habitat to avoid extinction, which has been called before in ecology a drift paradox, but there is no paradox, essentially, what we just observed here. Okay? And now, if we relax that assumption and there is no instantaneous death in the matrix, so the individuals can go into the matrix, they can wander there, and then they will return to the habitat. In this case, it makes sense to turn on a force of return, okay? So they realize that it's not very good a place, so they will try to go back to the habitat. This is the kind of situation that we get. The population distribution penetrates into the matrix like this, and we want to study what happens if we change the rate of return to the habitat, okay? And what we find is that there is a minimum for the total population size. If we plot the total population size, rescaled here, versus the rate of return to the habitat, there is a minimum here, okay? So if you increase the rate of return from a bad part, this is bad for the population. And this is because individuals return too quickly to the habitat, and therefore there is over-competition inside the habitat. So they compete a lot. There is a lot of people wanting to stay in the good place, okay? And then eventually, when we have essentially walls on the edges, when this rate is too large, you get that the individuals never die because they never go to the bad part. Okay, okay, so as concluding remarks, I have a few minutes. Important insights about collective behavior can be obtained by developing simple models. And we have found effects in active matter. Swimmers, unlike marine bacteria, move superdiffusively for much longer than simplistic analysis predicts. Motility heterogeneity can change agglomerate sizes dramatically. With motility-induced agglomeration, disease or information spread is optimal for intermediate persistence. And in ecology, spatial distributions depend non-trivially on attractive locations in fragmented habitats. And our model predicts how deforestation changes critical habitat sizes, being potentially relevant for policies. And now before I finish, I just would like to advertise this school and workshop on active matter that we are organizing together with Danilo and others, with Rodrigo Soto, Peter Solech, Julia Yeomans, Aparna Bhaskaran, which is going to be in early October. So if you know anyone interested, please let them know. And I finish with collaborators and funding. Thank you.
So any questions or comments you want to make? One.
So just in your ecological problem, you have the heterogeneous habitat. Is the diffusion constant the same in both kinds of habitat?
Yes, the diffusion constant is the same. The only thing that changes is the attractive force to return to the habitat or not, if you are inside or outside.
And what's the boundary conditions?
So essentially, you just go through the boundary. There is no actual boundary in terms of the... Yeah, in the first version, actually, we had absorbing boundary conditions because you would die immediately. But then you just go and changes the R, the net birth rate.
Do you have just one species, one density, or you can have competing species with that included in the internal dynamics?
That's a very good question. That's something that we would like to do at some point. There are some people with Celia Anteno, I never know how to say her second name, Celia from PUC. Exactly. There are some people who were here at ICTP and that worked with her doing something along these lines.
I have a question. So all these plots, they look very much like quantum mechanics, right? And the equation itself is similar to the Schrödinger equation. So the question is, if you have a good habitat, a matrix, and next to it another good habitat, can you have some sort of tunneling?
Yeah,
You
can talk about Anderson localization or whatever.
All that happens. Okay, yeah.
Yeah, I mean, in principle, I think you can. You are the perfect people to talk about this. You choose. Yeah.
Okay, thank you. So any more questions or comments?
Would be relevant to consider a situation where the diffusion constant is not constant, no, but varies. Now you have a heterogeneous habitat where locally or in a certain region you have some value of the diffusion constant, but it fluctuates around your habitat. Then you have to take into account, into consideration, fluctuation of the diffusion of the environment through the diffusion constant itself. So
yeah, there are models doing this. I think you have a model where when they go into the matrix, then the diffusion.
In real situations, sometimes the movement characteristics are different from habitat to matrix. And then this is translated to by having different diffusion constants, which creates a problem with the boundary condition.
Yeah.
They are non-linear jumps and so on.
Yeah, Gabriel, who was here, developed this. Yeah, so it depends on species, on the level of accuracy that you want to give, on the landscape itself. Yeah, but yeah, that's really interesting.
Okay, so thank you, Pablo. Let's thank him again. So next speaker now is Roberto Kraenkel from IFT.
Hello. Okay, so thank you for being here. So one good thing about complex systems sessions is that you hear about a lot of different subjects, actually. Yeah. So I will give a talk about modeling COVID-19 in the context of Brazil. Okay? So, and I will explain what's different when you say in Brazil. I'm not giving a talk about how to model in general. So, this is the 101 of epidemic models that probably all of you have, with COVID, everybody became a kind of modeler for in epidemics. So this is the simplest models that exist. Usually take your population, divide it in compartments, and then you define the flow between the compartments, and you get this kind of equations here, like the, is this working? Oops. Which is the simplest model that you can have. And then you have your epidemic curves here in blue is the susceptibles, and so in red the infected, and the recovered. Okay, so these are very simple models, and so like physicists we love simple models, but we have to be not too simple. So one thing what you can do with this kind of thing, okay? So one of the parameters that's important for in epidemiology is the concept of R0. At the very beginning of the epidemic, R0 gives you the number of secondary cases that one primary cases will create on average, okay? So this can be calculated from the models or even without models. And this could be made with simple models, but what is the use of this? So the use of this in this case is that R0 is connected to the total number of people that will have been infected at the end of the epidemic. So it connects the beginning to the end. And this is important. But obviously, things in real life are much more complicated. Why? Because with simple models, you are not modeling human behavior, which is, and human behavior will make your model much more complicated. And what you get by calculating the final size of the epidemic in terms of R0 is the worst case scenario. Nobody ever changed behavior. There's no government, anything, nothing happens. Just let the epidemic run. This is the worst case scenario, okay? So that to the famous calculations by people from Imperial New Ferguson, that you could have 2 million of dead people in US after the epidemic, okay? So this is the worst case scenario. But there is no such thing at, I think this is, yeah. There's no such thing at a society that does not do anything. Even depending on the government, there will always be some reactions, okay? So this is the epidemic curve for São Paulo, just to give you an example. This is the number of severe cases, which is the number of people that have been hospitalized per week. And you see that this is quite complicated thing. Many things are going on. Your simple model will not be able to explain such a thing, okay?
São Paulo City or São Paulo City?
This is, I think it's city. Yeah. This is city, yeah. And just notice there's a part in red here, okay? So I will now explain the part in red. So, okay. I will give you two examples of what you can do with models and data analysis that can be actually useful for policy decisions. So the point here is we want real data. We want models that use real data, which means that your model has to be adapted to the kind of data that you have. And then it comes to the point, we are modeling in Brazil. what is the data that you have in Brazil? Okay? So this depends pretty much on notification system. And the notification system in Brazil works fine for hospitalized cases. Therefore, your models, if you do a compartmental model, your model has to have a compartment of hospitalized people. But that's the only thing that you will be able, actually, to have. You have death and hospitalized people. You don't have, in general, you don't have an SIR model. The I is the number of infected people at that point in time. You never know. It's not given in the data. The data is the number of new cases of hospitalizations per week or per day. Right? So, okay. So now there's the concept of RT, which is the number of secondary cases created by a primary case at time t, at a certain time after the beginning of the epidemic. Okay? This can be calculated without any compartmental model. You don't need to use compartmental models. You can use kind of just defining the things. What you will need is to use this formula here, where I is not the I from SIR. I is incidence. It's number of new cases per day, okay, or per week. That's one thing that you usually get. If you're talking hospitalized case, that's the data that actually you can have. Okay? And then there's a parameter here, which is this W, which is the distribution of times. Say I'm infected today. How long will it take me to infect another person? Okay? This is called generation time. And this is a distribution, generation time distribution, which you don't have it because you don't know when you get infected. And you know what? I get infected yesterday. You know when you have first symptoms. Therefore, what you can maybe get from data is the data. It's a proxy for the generation time based on symptoms. And that's what people do in general. So, okay, so it seems fine. We have the data, we have everything, just calculate. Not so simple. Why? Well, we have severe cases. Now, every case has to be connected to a time. Okay? This case is connected today to the day yesterday or whatever else. So, what time is this? It would be the best if you could connect this to the time of infection. But you don't know the infection. You go, but you get the first symptoms. Okay? Because what we have seen in public discussion in Brazil is connecting to the time of notification. But time of notification is an administrative time. It depends on many things which are pretty much different from city to city. So the best thing is to connect to a biological meaningful time, which is the time of first symptoms. Okay? But then you have a problem, because your notification system has lags. It takes sometimes 3 weeks in order to, from the person that got the symptoms, then there are some days it goes to the hospital, it's hospitalized, then it takes still 2 weeks until this is somebody's actually typing in the system. So if you're now we are modeling an ongoing epidemic, not something that happened in the past. Your data is not consolidated. So you don't know the data today, you know only things that happened kind of months ago, which is not what the policymaker is wanting. In order to be useful, you have to say something if it's not about today, if it's less days. So what you do is you use the data, the full data sets that are available from the Ministry of Health, and you do an estimation with statistical methods, Bayesian methods, things that can get quite complicated, so that instead of forecasting anything, you are now casting how many cases you have now given that you know the past and given the first notifications that you have. So you can nowcast. So once you nowcast, then you can get a corrected curve, which is this. This is the nowcasted values, okay? If you don't nowcast it, it would be just going down all the time. You'd always be in a situation of a dying epidemic, which is not true. Because it's just a problem of the lag in order to notify. So once you get this correction due to nowcastings, then you can calculate the RT, okay? Which gives you a kind of strength of what is going on. Is it increasing? Is it decreasing? And so on. This is then you can do this, and actually we did it, and we put it on a web page, not on a paper, because a paper seemed to us in 2020 it was more important to put it in public, kind of public service, and instead of publishing a paper with that. Okay? So now, then there's a further point about this problem is there is the situation that people wanted to know this RT number, okay? But there are many situations where RT is not a good policy instrument. At the very beginning, R0 and RT are good policies because you have a lot of increasing. It gives you kind of speed of increase. You can know if is it slowing down, is it going up, how things are going on. But once you have a small number of cases, you have already been after the peak, you're in a moment where you don't have so many cases, RT will fluctuate completely crazy. And it is not anymore a good policy instrument. Okay? Because any small outbreak on background of 0 cases, any small outbreak gives you an RT. It doesn't mean that you had really a situation where you have an explosion of cases. So there's an implication how to use these results in policy. It is a part of policy information that is important for the people that will take the decisions, but it is not just I give you this number and you monitor this number and you define everything in terms of this number. This won't work. Okay? Okay. Now my second example is connected to this appearance of a new variant, which was called P1, and after that, at a certain point, called gamma variant. And this is the number of new cases per week in Manaus. And you have a super peak, which is in January 2021, which was a drama that everybody in the world actually has seen. Okay? So what we were interested at that point was how much effective strain, okay? The resident strain. Because it was very curious that in Manaus you could have such a situation because Manaus had a very high incidence before. And so you expected that the number of people that were immune would be very important. So 2 things is higher infectivity of the variant, and second, reinfections. So this has been discussed with the Ministry of Health because officially there were no reinfections because people didn't have data. Because they have to say something is a reinfection is a person that went through a PCR test and months later, this same person has also, again, a severe cases, and you get a PCR test and say, okay, then this is difficult to happen. And you don't have this name in these cases. So what we did, we wrote down a model, a compartmental model, which we call an intermediate complexity model. We have two different compartments. You have exposed to the wild variant or to the new variant. You have two variants. Then you divide people that get infected into hospitalized, asymptomatic, and infected without being hospitalized. You assume that people die only if they have been hospitalized. It's an approximation, which is pretty good. You get the recovered here, but these recovered from the first one can be reinfected by the second. Then you write down the model, and then what you need is parameters. Okay? So there are some of the parameters that are well established in the literature, but others not. And what we did is just parameter fitting for some important parameters. One parameter in every epidemiological model that you fit is what is usually called the beta, which is the infectiveness of your virus or bacteria. Okay? So what's the probability that if I'm infected and you are not, that after contact you will get infected? There's a probability, and this is difficult to measure. You cannot do this in lab. So this is a parameter that will always be fitted to the model. Okay? And the other things that we have fitted, it's the parameter measuring the number of reinfections, and also the initial number of people that already had been infected by the wild resident variant at the moment of the introduction of the new variant. Okay? So, and then we also needed to have results about the proportion of the new variant with respect to the old one. Okay? That was also available at the time. And then what you get is that the new variant is about 2.6 times more infective than the wild one. But and you had a prevalence of the previous infection of 78%. So this model shows that it's compatible, that you had a high prevalence of the wild case, but this is compatible with a reinfection scenario through a new variant that's much more infective than the previous one. Okay? And then these results, okay, that's what I just said, but we also estimated that the number of reinfections, the total number of cases between the beginning of November 2020 and the end of January 21, all the hospitalizations for COVID-19 in Manaus, 28% are reinfections. And surprise, surprise, we had people that actually can measure that. So people use, it's a proxy, blood donors. But there's the blood of the people, and then they can go and see if you have reinfections. And they actually found 22%, which is in our the range of values that we actually, I didn't write here, it's 28 plus or minus something. Okay. So this is an example of using models not to do what people usually would first think with a model, to predict future. We have seen that predicting future for epidemics is much more difficult than you can ever think when you sit on a computer lab and try to do some simple model. Because there are variants, there's behavior of people, heterogeneous of all kinds. But you see, even this is not a very complicated model. It was a it is also age stratified, okay? You have to, but not very conceptually easy to understand. So this is what I call an intermediate complexity model. is useful, but it's useful not to predict things. It's useful to understand what is going actually on. Infections, infectivities, and this kind of thing. Okay? So, and that's it. Thank you.
Danilo.
I I have a question about your data in general. Do you have any parameter to measure, for instance, how severe parameter that measures how severe the infection has been, for instance, available for you? I know one extreme case would be the number of deaths, for instance, but do you have something?
We have only the person has been hospitalized. So you could, we didn't do this, but we worked together with a big group from Oxford, doing a kind of model that could be adapted to many places in the world, where you have, instead of having only infected, hospitalized is hospitalized and hospitalized that need a respirator. So, but this is not in our Brazilian data, okay? So we cannot use it. I mean, so there are people around the world that use this. They hospitalize people that need special respirators, or then there are people that need respirators but don't get one, and so on. You can introduce this and go on. The point is, can you connect it to any kind of, what is on your data?
Of course, we want to do predictions, but we can also learn a lot, especially during all this negationism and etc., looking in the past.
Yeah.
So, could you find common features? Because besides these numbers that you have, you also have, you can make the look at differences from looking at cities, states, regions, what they have done and what they not have done in terms of since vaccination, isolation, all those things. Because when they analyze the world, they took this measurements and wow, that was a big improvement in terms of number of deaths, etc. So in Brazil, were you able to look around and use the data in the public measurements they took to make a distinction what worked, what not worked?
Okay.
I think that would be extremely helpful to exactly to say, well, you see the numbers can, that would, I'm not talking, the epidemic is over, but we could learn a lot if we could somehow classify, clustering different similarities of what is the outcome compared to what people, how they behaved, etc.
That's the kind of thing that people actually do work with. We have also done, for instance, evaluating school closures in different states of Brazil and so on. Well, then there's always the problem of the data, but the data is, for hospitalized people, it's a public national-wide data, and it was open, without the names of the people, okay, without identifications. But then there are, for instance, data of school closures or openings, or partial openings, are collected through public media. So it's kind of, you depend on, so we have worked with people in our group, as persons that do political science and on quantitative methods. And these people go on and they examine all the rules that are applied in a certain place and so on. So you can have more or less. And then you can do this, what worked better and so on. Are there nonlinear effects that are important or not? So lots of things are being have been done, and are still under analysis of what's worked or not. For instance, one thing that we did was calculating how many lives were saved due to vaccination. During a given period of time, from the start of the vaccination until August 2021, which is when the vaccination started to go below 60 years old. So we could actually find a projection of how many people actually didn't die due to that vaccination. Obviously, you can also say, if we started earlier, how many more lives would you have saved? So this is also things that you use. In this case, the data is historical in the sense that you don't have this problem of now testing and so on, which is when things are ongoing. So this is also an example. I think there's an...
Did you try your method or model with other city? Question one. Question two, is it depending on the density? Meaning, a very low, dilute system would have a different behavior than a dense system like São Paulo or even Manaus that is not so dense, but the people have this outside behavior. Is it dependent on the model or not?
So two things. We did it for all Brazilian states and all capital states and some big cities which are not capitals. It can be adapted, but very small populations will be terrible. Your error margin will be super high and absolutely useless to apply. Okay? But we have done this from on the site. The site is not anymore there, but for all the states and main cities, and the density. So that's a point about density is in your compartmental models they are always frequency dependent, not on the total number of the population. This corresponds to the pattern of human behavior, which is, say, we have 10 million people here, and Campinas has 1 million people. But they proportionally, the epidemics are very similar. The duration of the epidemic, everything is similar. It's not that in São Paulo, you would have higher densities or more people. Okay? So these density effects are usually marginal in epidemics. In humans, because where does it come from? How many contacts do you have in Campinas and how many contacts do you have in São Paulo? It's essentially the same because it's come from human society, which is not true, for instance, if you're doing epidemics with animals. Then you have.
Okay, thank you, Roberto. Yeah, I know we all have questions about COVID, but time is running out. So next speaker is Danilo. Danilo is the last speaker of this block, but don't go away, there are more talks coming next.
Okay. Okay, I think I can go on. Thank you, Marcos, for the introduction, and thank you all for coming. It's a real pleasure to give this talk here. It's really a very nice gathering. And I echo Roberto's comment that you're talking about complex systems. You can talk about almost anything. And today I'm going to talk about this ubiquitous criticality of disordered solids. And I'm going to start with a transition or a kind of system that is very paradigmatic of disordered solids. And I'm going to use this so-called jamming transition as a proxy to introduce many concepts that I'm going to use and try to argue how we can see some universal behavior in lots of different systems like colloidal gels, shear thickening, and even strange metals and things like machine learning. So here, after I introduce jamming a little bit, I'm going to talk about how we can come up with very simple modeling for these systems, just mass spring systems, for instance. And then I'm going to explore these results and try to shed light into some new experimental results for systems very different, like strange metals and shear thickening and things like that. So let me start with jamming, and I'm going to start with a very simple situation where you have a bunch of disks, for instance. They might be hard disks, they might be soft disks. I'm not going to pay too much attention to the particular interactions that they have. But the point that I want them to be polydispersed in size so that when I compress them, they don't crystallize. And I'm going to apply some isotropic compression on this set of disks that's polydispersed in size or something like that. And what I expect to happen is that if I have compressed that for long enough, I'm going to have form a disordered contact network for these spheres, and the system jams. It resists compression, so that's what happens. And you can describe this using, for instance, elasticity theory. You can look at the elastic moduli. You have a bulk modulus, you have a shear modulus for these systems, and right as you ramp up the compression, the bulk modulus jumps here to some finite value, whereas the shear modulus starts growing continuously from 0. That's a kind of an odd transition, and if Ehrenfest were alive, he would be kind of puzzled if this is a first order, if this is a second order transition. But that's not what makes jamming special. Is that jamming is important because it's universal. So it's universal because it shares this common structure with several different disordered elastic systems. And I'm mentioning here some of them, like the jamming of spheres that I've just told you about, but also confluent cell tissues, even systems outside condensed matter, like machine learning, and strange metals, for instance. And I put universal here between quotes because universal, you know, we know what universality is in the context of statistical physics, and mainly due to this, to the work of many people, including these three gentlemen here, Ken Wilson, Michael Fisher, and Leo Kadanoff. And we know that in a critical transition, there is some sort of fixed point in a renormalization group transformation that controls the criticality of the system. And you can define a number of, a relatively small number of relevant fields that you set to particular values in order to get to the transition point. And there is a critical surface here where all the relevant fields, that might be infinitely many, all converge to this same fixed point. But the point is that there is a fixed point that controls the criticality of the system in general for this. And what we expect is that there is some sort of jamming fixed point that controls the universality in some sense for these very different disparate systems. And it would be nice to have some sort of renormalization group approach that would explain all these results. Unfortunately, today I don't know of any successful renormalization group approach to jamming transitions. But what I'm going to do next is to try to, by assuming that jamming is universal, I can come up with simpler model, leverage the supposed universality, and to explain the jamming transition in some sense, and also use the results that I obtain with this simple model to address experimental measurement for other different systems. So that's what I'm going to do for the rest of my talk. So what's the simple model that I'm considering here? And I'm going to model jamming as some sort of disordered elastic network. And I cannot go to all the details. And if you ask me or if you want to chat, I'll be happy to chat about this in more detail. But what I have here is a combination of two lattices, a honeycomb and a triangular lattice. And what I'm going to do, I'm going to consider springs connecting those nodes in both lattices, and I'm going to populate each bond on each lattice with some probability. A probability PB for the blue lattice, and a probability Pn for the red lattice, for the triangular lattice. Now, depending on the probabilities, you can have different types of transitions where you lose rigidity or not, or something like that. Now, another thing that I'm not going to go into the details is that I'm going to use a mapping from this diluted network system to a homogeneous effective medium system. This is based on an approximation that goes by the name of coherent potential approximation. The name doesn't matter, but we have this methodology that we can use to to map the diluted system into an effective medial system. And by the end of the day, what we get are effective spring constants, a homogeneous system with effective spring constants, KB and Kn, that will, in general, depend on these probabilities, PP and N, for instance. So that's what we do. And you can solve the equations that you get for these effective spring constants, expand the equations near the critical point, near this jamming critical point, and what you get is something like an effective spring constant that will depend in general on the excess contact number, for instance, the delta Z, that's the parameter that controls the transition there, and potentially also in some frequency omega. Now from here, I can calculate for this system with this effective spring constant what the susceptibility is. This is a displacement response to a force. So apply some force here, and you measure the displacement response to this force, and that can depend on the wave vector and can depend on omega, and I can derive, using this theory, some scaling form for this displacement response, which is a susceptibility. It's a very fundamental quantity. And here we get something like the critical exponents, gamma, nu, and z, and also some universal scaling function, curly L. And just to give you a brief reminder of what I'm talking about here, I'm going to use the example of the Ising model that is familiar to most of us here. And you have a phase diagram like this with field and temperature and a first order transition line that ends at a critical point. And in general, thermodynamic functions will have some sort of generalized homogeneity relation like this that can be derived, for instance, from the renormalization group approach. And from this, you can choose the scaling factor B to eliminate one of the variables and get something like a scaling form like this. So the magnetization as a function of field and temperature is not a function that's arbitrary, but it should be some sort of power law times some universal function of this invariant scaling combination here. And what we have here, I cannot see because it's on the top there, but I can push it back here, is just the analogous thing, but for this susceptibility. Now, with this theory, we are able to not only obtain the critical exponents, but also an explicit formula for the universal scaling function. And I'm not going to give you the details on what kind of assumptions I'm making here. I'm just talking very general about this. But we can write down the explicit formula for the universal scaling function. And it's given by this in this case. And I'm going to skip it for the interest of time, the explanation of what this plot means. But let me just make a brief comment about the value of extract information about the universal scaling function. You know that from the theory of renormalization group, not only the critical exponents are universal, but you have your other quantities, like for instance, the ratio between critical amplitudes and things like that that are also universal. And if a function like this is also universal, it becomes as important to know the function as it is to know the critical exponents, for instance, insofar as critical as universality is concerned. So there is a point, there's an important point on finding the universal scaling functions, and that's one of the lessons I'd like to bring home today. Yeah. So I mentioned that we use this susceptibility, and it is a fundamental quantity from linear response theory. And from that, I can extract the scaling forms for lots of different quantities, like elastic moduli, viscosities, correlation functions. In particular, I can look, for instance, from here, I can go to the structure factor of the system, and there will be a different exponent here, a different universal scaling function here, that are related to these other functions and exponents, and I can plot this. I can, for instance, rescale the structure factor here and plot in terms of rescale the wave vector and rescale the frequency, and the plot looks like this. And this shows, for instance, this rounded maximum here for fixed frequency and almost independent of the wave vector. That for those of you who know glass literature, this is the boson peak that's related to glasses and systems near a jamming transition in general also. And I'm going to mention this just because I'm going to make connections between this and some experimental measurements for dynamic response functions for strange metals that also show the same signature of a kind of a boson peak here. So now I'm going to mention very briefly some systems that exhibit jamming-like transitions and try to argue for this universality of this jamming transition for these disordered solids. And I'm going to start with the strange metals. And this is a strongly correlated system, and I'm not going to give you all the details about this. I'm showing the phase diagram here for a high TC superconductor. This is one of the famous cuprates. This is BISCO. And I'm just pointing to this SM phase here, which is a strange metal phase. It's strange because it exhibits lots of anomalous behavior from what you would expect from Fermi liquid theory. One of these anomalous behaviors is, for instance, you can observe in measurements for the dynamic charge density response function as a function of frequency or energy, if you like, for different values of Q. And here again, you observe this rounded maximum almost independently for a certain energy scale that is almost independently of the wave vector. And that kind of reminds you of the boson peak of glasses. So what we did next was to try to add ingredients in our random elastic system to at least qualitatively reproduce some of the results that are observed for the strange metals. And we added, for instance, Coulomb interactions using some approximation that is also known, the random phase approximation and things like that. We also added some heterogeneities. And in some sense, we were able to reproduce some of these features that were present in these experimental measurements. Of course, our model is not a direct model for strange metals. It's not quantum. It doesn't have lots of the ingredients that you would need to actually describe a strongly correlated system like this. Instead, what we want to point out is that there might be signatures of jamming-like transition in those strange metal systems, and maybe provide some sort of new window to look and interpret the experimental data for these systems. So I'm going to briefly mention also rigidity percolation. I could have started with rigidity percolation because this is well known and well studied, and it's a picture that's much simpler than the jamming picture. You can, for instance, start with a triangular lattice and remove bonds of the triangular lattice, and what you would get is something with a phase transition at some critical coordination number where both bulk and shear moduli grow to 0 at the transition. And our model is capable to explain both, the jamming transition and the rigidity percolation transition. And jamming emerges as a kind of multi-critical point terminating a rigidity percolation line. Now, that leads us to conjecture, for instance, that there would be some sort of flow diagram where you can describe some jamming fixed point here, rigidity percolation fixed point, and we can extract information using something such as a crossover scaling theory in order to interpret and to explain these results. Now, I do not have time to go into the details, but we also used the same kind of idea to try to understand some experiments in which there is a shear thickening transition. For instance, you probably have seen some movies where people walk on the water. It's not water, it's water. It's a colloidal suspension with cornstarch, for instance, or something like that. And people are able to walk. It's totally a fluid, but people are walking on it. And the reason for that is that when you apply some stress on the system, the colloidal suspension thickens. The viscosity goes up sometimes by an order of magnitude or so, and it might be also discontinuous. And in one of our studies, we connected this with the general flow diagrams, for instance, for isotropic jamming, shear jamming, and it turned out that those critical transitions kind of were also responsible for this shear thickening behavior, and we could fetch data, collapse data, and things like that. But I'm not going to give details. If you're interested, I can discuss more about this data. So that's it for my talk. There are a bunch of relevant publications here, and if you, I'd like to thank my collaborators again and you for your attention. Thank you.
Thank you, Danilo. Any questions, comments, Marcia?
Fascinated. You see, many years ago, I work on phase transitions, kind of recall ancient time. But when you describe with the critical exponent, you start with susceptibility, but if you go all the way to the top, you're saying that the free energy of your system will have a regular part and a singular part, and that this singular part knows that when you do a certain process, because when you go glassy, when you go jamming, is one path that arrives here, but if you take another path, you might arrive someplace else. So you envision families of ways to write your free energy with this regular and singular part? Because you understand, you don't have, you can have many glasses depending on the process. You can have many jammings if depending on the process. So you imagine that you can somehow rewrite your free energy depending where you are looking in your phase space. Okay, so I'm looking here, it's telling me that I'm going there. If I look here, there, looking at the free energy level, or you don't want to go there?
Yeah. That's an excellent question, and these systems are tough to do any sort of renormalization group approach or anything that would resemble a renormalization group approach. I envision trying to go by this using networks, which are simpler systems, and maybe they are more controllable in some sense. They are still difficult and they are still toy models in some sense, that it's hard to, there will be new interactions that will be generated by the renormalization group approach, but they are still into this family of random elastic systems instead of the more general family of spheres touching one another or something like that. So I wouldn't know how to go about this more general approach. But for disordered analysis, maybe, yeah.
Any other comments?
I suppose if you perturb your system in the jamming transition, you can cause it to end jam, right? You can, yeah. And then if you keep it the perturbation on, you might have near jamming transition, then it unjams, and then you might have a quite interesting dynamics there. Have you studied that? I mean,
I particularly not, but there have been many papers on unjamming transitions by lots of different mechanisms. One of the mechanisms, for instance, that I've been exploring and been looking at, that I talked with Pablo, for instance, is adding activity on the sites of the random elastic network. And there have been some people who studied the unjamming transition that is brought about by the inclusion of self-propulsion in these systems. So definitely it's interesting. There has been lots of work done on this line, and yeah, I'm interested in this one.
Okay, we have time for one more question, if anyone.
So how important is polydispersity? Because you diluted the system, right? So the system, because of your stochastic dilution, is a polydispersed system in some way. So it's heterogeneous, right? So, And then you mapped this onto a homogeneous system. But if you diluted the system and kept it homogeneous, so you would have a lack of bonds, but still it would be a homogeneous system. Would the exponents change? Because for colloidal mixtures, if you have either a mixture or not, it changes the exponent.
No, let me try to break the question in two parts. For instance, the polydispersed part for spheres, it's central, because otherwise, usually, as you apply some sort of protocol, the system crystallizes. So it forms a crystal instead of this disordered structure. But what polydispersed system are you thinking in terms of the random elastic models? The fact that I'm diluting the system or removing bonds or something like that?
Yeah, exactly.
There have been other approaches that do not need the dilution of bonds, and that you can also employ based on random networks. You can use the random networks instead of using those regular networks that I've shown, and that give the same result. One ingredient that you must have for jamming is that your network has to have some sort of structure that resists compression. It has both things, that resists compression, and it's right at the isostatic point, the limit of stability. So if you are able to mimic this, we have shown also that you can do that without all this protocol of adding or removing bonds or things like that.
So in principle, if you dilute trying to keep some sort of homogeneity or not, doesn't matter for the critical.
No, not necessarily, yeah. It has to be disordered because the structure is disordered, but yeah.
Okay, so thank you, Danilo. So we have a short break now. We resume in 10 minutes, okay, at 3 o'clock.
Eu acho que eu falo às 4h15, se eu me lembro que fala.
Eu imagino que isso seja você tem que programar a rede.
Eu falo assim, porque no final você ganha uma rede irregular de novo, só que uma rede irregular é
dar oi pra você. Olha, eu falo quase com você, mas vou tentar fugir. Assim que eu acabar, eu vou tentar correr pra cá. Numa outra aí. Eu sou a segunda e você é o terceiro. Eu vou tentar chegar até você. Tudo certo?
Vamos aqui, tá? Eu vou um café provavelmente. Já está aqui vendo os seus posts.
Agora o CNPq está tomando conta da minha vida. Não tenho conseguido fazer tantas coisas, mas estou tentando.
Mas você está bom.
Eu estou em Brasília. Assim, uma vez por mês, eu faço parte de um negócio que chama CGIBR, Comitê da Governança da Internet do Brasil, que eu sou indicada do Ministério. E essa reunião é em São Paulo, uma vez por mês. Aí é uma sexta-feira sempre. Aí eu vou para Floripa e fico lá na segunda. E volto na terça. Então é o dia que eu tenho lá. Então é um dia por mês. Mas é sim. Iniciação científica me livrei de todo mundo, inclusive uma vindo aqui trabalhar com o Kraenkel. Mestrado, tenho uma acabando e não vou pegar mais. Doutorado, tenho uma acabando e aí já disse 2 nomes porque eram pessoas que estavam mais distantes. Então eu tenho 3 pós-docs, aí dá para tocar. Mas aluno nem sempre possível. E mestrado também, se for muito cru. Não, eu estou fazendo umas coisas que eu estou adorando. Eu estou por ordem, eu tenho os dados lá. E a equipe técnica é muito boa. Então eu tenho ideias e eles vão revelando, vão fazendo os painéis. Porque precisa avaliar alguma coisa, primeiro tem que ter os dados organizados. Não é uma mudança de cultura no CNPq. Então estou gostando, é um desafio. E está saindo resultados. Agora a foto é grande. Tem que melhorar muito isso. Eu vi o final, mas eu passar é muito bom. O CNPq tem uma verba esse ano menor que a do ano passado. Porque no ano passado teve um pouco do dinheiro da transição que entrou para a ciência. Esse ano não tem. E como teve aumento de bolsa, de lá, sobra pouco dinheiro para fomento. Mas a expectativa é que as pessoas tenham paciência em 2025 melhor.
A gente lá em Porto Alegre não é assustado, porque já deu vários. Tá feia.
Os meses lá foi danado.
Não. E assim, vai ser isso de agora em diante. Assim, a gente tem que se preparar. Tem gente que se acostumar. O mais divertido, gente, é que os senadores estão chamando reuniões para falar sobre essa catástrofe. Os senadores bolsonaristas, entendeu? Chamam, se reúnem, preocupados com as mudanças climáticas afetando o sul. Assim, não, me belisca. Eles chamam para explicar. Me belisca. Assim, falar da mares, todos eles, assim, eles não estão nem aí com a seca da Amazônia. Eles estão preocupados com enchente, que é absurdo isso, que está sofrendo a população. O que a gente vai fazer? Eu tenho que fazer cara de... tudo bem, a gente está fazendo isso, talvez... E vocês negavam isso. Exatamente isso. E assim, Santa Catarina não é bem água que está batendo. É mais grave ainda, porque resolveram fazer. O pessoal fica torcendo quando eu vejo essas coisas. Que não tenha dado aula para os engenheiros que fizeram essas obras horrorosas que estão em Santa Catarina, que fizeram aterramento que vai até o oceano. E não entendem que assim, a gente já está em Porto Alegre no terceiro tornado. As pessoas reconstroem igual como tinha um mosquito, sabe? Vai ter de novo. Não, mas isso não, eu não posso ter dado aula para essas pessoas. Não é desesperador. E no sul não está pegando essa bola. Então isso é mais desesperador.
Pega as vasilhas ali para dentro.
Não, assim, a cidade, na parte, assim, pega bem no moro, na cidade, que é perto do rio. O rio Guaíba já foi aterrado. O estádio, se não se estádio de futebol, totalmente em cima de aterramento. Totalmente em cima. E shopping center em cima de aterramento. Eu olho aquilo e digo, pensando, tomara que os engenheiros que fizeram isso sejam melhores dos que aterraram agora a praia, mas Santa Catarina.
Lá em Recife, vez ou outra, fazem isso também. Aterrar.
Vocês ainda eles estão na região de dupla, porque assim, ó, lá em cima vai ser seca, aqui embaixo vai ser. Vocês estão quase na região que começa a dupla. Uma parte sudeste tem uma dúvida se vai ser seca demais.
Look at, that means that something enters the nucleus, enrichment of this transcription factor. So these are figures from the paper where they presented the results of this experiment, and what they observed is that not only the transcription factor differs in its dynamics depending on the stimulus, but also the way that the stimulus intensity is encoded in the properties of the nuclear fraction of the TF is also different. And so what they found was that when they are subject to osmotic stress, the intensity of the stress, which is here, let me see, here, is this, in this axis here, is the duration of the pulse of the transcription factor increases with the intensity of the stress. If you apply oxidative stress, then the amplitude of the this enrichment of the transcription factor in the nucleus increases with the intensity of the stress. And if you have the glucose limitation, then the frequency also increases depending on how much you deprive the bath from glucose. So there are three codification schemes, and this you can call dynamic multiplexing of the information transmission. So dynamics allows you to use the same transcription factor to transmit, to signal and identify different types of external stimuli. Then this is another example with yeast, and which is of different nature in the following sense. Here, when you subject to a food deprivation via the cells, there are two types of cells that appear, and each of these types of cells, yeast cells, MAT-A and MAT-alpha, they secrete a pheromone that attracts the other subtype, so that they can, when they are subject to starvation, then the two types secrete this pheromone and they start to grow towards one another so that they can fuse and produce a new cell that is able to sporulate and propagate the species. And so in the experiments, what you do, you cheat the cells. Basically, you get one of the subtypes, is MAT-A, and you add the pheromone, which is called alpha factor, and then you see what the response that happens. And well, the whole signaling pathway has been dissected, and the changes in gene expression you see as what new proteins you produce that you observe in the microscope. So now in this case, you have this polarized growth of the cell, and so what you have to detect is not just to distinguish whether the pheromone is there or not, but grow towards the region of largest concentration. So it's a detection of the gradient. And this is a figure in which you see all the cells growing in sort of in the same direction, which is the direction of a larger concentration of the pheromone. And so how do they ponder these different concentrations? How well can they do that? Well, experimentally what you do is subject the cells to different concentrations, and these experiments have shown that putting this pheromone in the bath induces either a stationary type of response, but also a pulsatile type of response. So what is the use of these two dynamics? What's the advantage? And so in the previous example, we had one transcription factor with different dynamics depending on the stimulus. Here we have the same stimulus that can induce different types of dynamical behaviors along the response. And so we try to answer, to go into these questions and try to understand why this happens. So let me show you again this example of the alpha factor with yeast. This is increasing pheromone concentration. This is the level of response, which is stationary. This is the canonical way to respond. And what you see is that the response increases with the concentration of alpha factor, but also with increasing the concentration of alpha factor, which is the different colors here. What you see is that the cells show pulses of calcium, and the frequency, how many pulses you can elicit, increases with the concentration of pheromones. So you have a pulsatile and a stationary type of response. So you have what we call amplitude encoding and frequency encoding. Anyway, so the way that we approach these questions got inspiration from Bill Bialec's work. Basically, we work with mutual information. And so this is based on the assumption that the environment can be in many states. Well, we sort of count them, the states with a certain probability, and then the cell obtains data from when the state of the environment changes, and then how much information can it gain depending on the response that it generates. So you compute the mutual information between something that works as an input and something that works as an output. And this is the expression of the mutual information. And so what we wanted to do was how the mutual information could change depending on the different types of codification schemes that we saw in these examples. And we first compared, for the first example, that showed that the same stimulus could produce different dynamics on the same transcription factor. Different stimulus could produce different dynamics. What we looked at was our input was the transcription factor, and our output was the messenger RNA. And to work with the other example, the same stimulus producing two different dynamics, then we went from the external stimulus to the messenger RNA and computed the mutual information. Anyway, so let me tell you a little bit about what we learned from their first work. we used a very simple transcription model that we took from a paper by the people who had done the experiments in yeast. And we computed the mutual information between either the amplitude of, so we went from the nuclear fraction of the transcription factor to the messenger RNA that was produced. And so we computed the mutual information between the mRNA accumulated over a certain time and either the amplitude of the transcription factor, the duration of the nuclear fraction of the transcription factor, of the mean interpulse frequency. So we did not include anything about the external stimulus here. And that was our input. This was our output. And for amplitude modulation, we assumed that the transcription factor had one pulse, so always the same duration, and the amplitude had a mean plus some noise. Then for duration, we assume also one pulse for various durations. And for frequency modulation, we assumed pulses separated by a frequency, mean frequency that was exponentially, so the time was exponentially distributed with certain mean frequency. And so what we did was we tried to find the parameters of the transcription model that maximized the mutual information for each of these codification schemes. And in particular, what we were after, we wanted to see whether those parameters values that maximized transmission for amplitude encoding were in a separate region of parameter space of those that maximized for frequency encoding or not. And we found that they were more or less on the same region. And then what we did was we looked for parameters that maximized information transmission for one encoding, and then within that region looked for those that minimized as much as possible for another mode. And so in that way, we defined 6 regions because we were comparing the 2 modes in pairs, duration and frequency, duration and amplitude, frequency and amplitude, and maximizing the first or the second. So we have 6 combinations. And then we picked one set of parameter values in each of these regions. And one set of parameter values would mean one promoter. And we wanted to see whether we could separate those that responded correctly to frequency from those that responded correctly to amplitude or vice versa. And what we found was that actually we looked at how much information these parameters, for these parameters the whole system could transmit. And so here in colors are the different parameter sets, so the promoters, which are 6. And then with symbols, different symbols, is how we computed the information, whether we computed it for duration, that's circles, amplitude is crosses. The other cross is somewhere here, I cannot see it. And the frequency is the triangles. And what we found was that duration, for duration transmission, the information was more or less the same for all the promoters. And the most sensitive one that could change more with parameter changes was frequency, that could be below 1 or almost 2, depending on the parameters. And so on multiplexing, our conclusion basically is that you can find promoters that are blind to frequency and transmit relatively high information for the other types of inputs, but we didn't find the opposite situation. And this agreed with some experiments of the same authors that looked at 2 genes that under physiological conditions responded to either frequency modulated signals or to duration or amplitude. And they observed that they more or less obtained the same mutual information for the one that was responding to frequency, for frequency and for amplitude, but the one that was better for amplitude did not transmit well information for frequency. And also they mutated these genes, and they found that they could increase the information, they mutated one of them, the one that transmits for amplitude, and they found that by mutating it, they could transmit more information. And so the authors were wondering why it was not optimized. And basically, our simple response would be, well, maybe it's not optimal because in this way it's as blind as possible to frequency modulation. So I don't know how much time I have, 5 minutes. So let me give you briefly the other kind of dynamic thing. We worked with the same transcription model, but this time we included the external stimulus. And so we computed the mutual information between the mRNA produced and the intensity of the external stimulus. And again, for amplitude, we assume a single pulse. And the thing that we had to introduce was how the amplitude of the single pulse would relate to the external stimulus strength. And we used a Hill function, which is typical of this, like the canonical response in yeast. With the strength, you have the intensity of the response goes like a Hill function of the external stimulus concentration. And for the pulsing encoding, what we picked was something that we got from observations of calcium pulses, which is they are separated by a stochastic time that has a fixed quantity. Usually it's like some recovery time in between pulses, plus a stochastic time that scales exponentially with the external stimulus. So the mean interpulse time is exponential with the external stimulus strength. And these two dependencies on the stimulus strength give different, qualitatively different response capabilities. For the external stimulus strength, well, these are the justification. Let me go through this, which is for the Hill function is clear, and for the interpulse time, as I told you, we got it from calcium signals, but actually we usually found this dependence for a nuclear localization of a transcription factor, particularly crazy in yeast. This is a figure from this paper, very old paper, but if you put it in log scale, what you find is that the frequency scales exponentially with the intensity of the external stimulus, which in this case is the extracellular calcium concentration. So, and this you can derive also from an excitable model, the exponential scaling in the presence of noise. Anyway, and for the external stimulus distributions, what we wanted to see was whether one or the other encoding mode worked better depending on the range of external stimulus strengths. And so for that distribution, we tried basically a beta distribution which has compact support, and by switching the parameters, you can move where the median is. So the range of external stimulus strengths that you are like looking at, basically. So these are the distributions, and what we found for amplitude encoding and frequency encoding, let me look at two of these figures. These figures have, in the Hill function, the same EC50 and different Hill factors, the three figures. And this is for frequency encoding, and this is the median of the external stimulus distribution. And what you see, and this is the mutual information, what you see is that the mutual information for amplitude encoding decreases with the median, or has a maximum which is sharper as you increase the Hill coefficient. While for frequency encoding, you can find parameters for which this remains relatively flat for all medians. So each of these symbols corresponds to a distribution with a different median of external stimuli. And so this is what I just said. And so the two dependencies are very different, and this is because of the different way in which the external stimulus the other one that use frequency encoding. Well, our assumption is that with the combination of both types of encodings can enlarge the range over which you can detect concentration gradients properly. So that's basically something that we are planning to analyze experimentally with yeast. Anyway, so let me summarize a little bit. Cells use different strategies to decode the information they receive from their surroundings. And sometimes they encode the increasing intensities of the external stimulus into increasing levels of response in a stationary sort of way. Sometimes you elicit some type of dynamical behavior with pulses, and the frequency between pulses increases with. Why is this? Because you can multiplex the transmission for what else? And so for transmission multiplexing, we looked at the mutual information between the transcription factor and the mRNA, and what we found was that we could find promoters, so transcription parameters of the transcription model that were so that the whole process was blind to frequency modulated inputs, but they transmitted very well for the other modes of encoding the external world, and the reverse situation we didn't find. And so frequency encoding is more selective. And then in the case of one stimulus and two dynamics, what we did was look at the mutual information between the external stimulus and the accumulated mRNA, and we saw that because of the different mathematical way in which the intensities is mapped to the property of the transducers of the signaling, that this is the intensity of the external stimulus, the way in which it is encoded, the transmission capabilities are different from a qualitative point of view. Amplitude encoding is optimal for a limited range of external stimulus strengths, and frequency encoding can work well for a much larger range. So that you can expect that the use of the two types of encodings can serve to enlarge the range over which the signaling works. And with this, I thank you and good day.
Thank you. We have time for one question, maybe?
I might have missed, but is this mechanism valid for any type of cell? Like if you have, stem cells and you try this, the mechanism is there, like a basic, IC mechanism that's already, that you try to model everything, or depends of the type of cell that you're using?
What I don't understand what you mean by
This
one way of producing responses is by altering gene expression.
yeah.
And you need to change the concentration of some intermediaries of the signaling to change gene expression. So in that way, that's a common way to do this. Now, whether
Because you classify between different types of signals that you observe. Is this universal, or if I change the type of cell I'm using might be different between the different types of signalization that you get?
you can have this type of signaling like pulsatile, or for example in transcription factors, nuclear localization of
Yeah.
transcription factors, a pulsatile or a amplitude encoded in different types of cells.
Okay.
Like for example, also in cells of mammals, P53, which is a transcription factor, also shows pulsatile dynamics or some sort of stationary, so it uses amplitude or frequency encoding, depending also on the type of stimulus that you're applying to the cells. Now the particularities of the time scales involved,
Mm-hmm.
that will depend on the pathway and the cell type
Okay.
Just out of curiosity, your output was single sigmoid with some Hill coefficient, but the mRNA comes on bursts. Do you have some comments about different size of bursts as this thing get modulated or not?
We didn't look at the dynamics of the mRNA. What we looked was at the accumulated mRNA, how much they produced over time. Now, whether the pulsatile behavior is related, the pulsatile behavior of the transcription factor is related to the bursts or not, that's a different question. But the burst of a transcription can occur if the transcription factors are not pulsatile. But we didn't look at that in this work.
Thank you.
Okay, we're running out of time. Thank you again, Silvina. So our next speaker is Professor Marcos.
So different type of talk now, very theoretical. Complicated title, but simple talk. Okay, so I think I have also this type of picture. This one shows a very large flock of birds that behaves collectively as if they were a single animal, like a dragon flying in the sky. So this pattern that you see here in space, it also happens in the time domain. So if you have a group of oscillators, and these oscillators synchronize their phases, these oscillators will also look like a single individual, a single big oscillator pulsing in time. So the classic example here are not birds, but fireflies that under certain conditions can synchronize their flashing. And over this past maybe 30 or 40 years, people have realized that this type of phenomena, synchronization of oscillators, it happens in many places in nature, not only fireflies, but cells in our heart have to synchronize. There are groups of neurons that have to spike together, power grids working in AC. And out of these examples, I selected one to show in a bit more detail, which is very interesting. So these are sperm. They are not human sperm. They're sperm from a sea creature. I think it's a sea urchin. And when the sperm swims, its head moves up and down. So this is an internal oscillator of the sperm. And what these people found is that if you take this sperm and put them on a two-dimensional layer of water, what happens is that they form these vortices that have on average 10 or 7 sperm. So the sperm make these vortices and synchronize the motion of their heads at the same time. And if you look at the large scale, you see lots of these vortices, and they form this type of hexagonal patterns. And there is more. People found recently that if you look at the tail of each of these sperms, there is a number of cells there that are responsible for the motion of the tail. And these cells have to work in synchrony, otherwise they don't swim well. So you see that if you want to describe this type of complexity, you need models that have several variables. You need several degrees of freedom that interact and will eventually synchronize. Spatial degrees of freedom, internal degrees of freedom, and this is a bit of a challenge. There is one very nice model that couples spatial and internal degrees of freedom. It was proposed by Strogatz. It's called swarmolators, things that swarm and oscillate at the same time. I'm not going to talk about that today, but what I want to describe to you is a model of synchronization that is a generalization of the famous Kuramoto model that I will show in a moment, that can be generalized to more dimensions to include several internal variables that will eventually be coupled to spatial degrees of freedom. So let me spend a few minutes talking about the Kuramoto model. It's a very simple model of synchronization and therefore very beautiful. It describes oscillator by a single phase, so there is a single variable. Each oscillator has a natural frequency, omega, and they are all coupled together with a single coupling constant, the same coupling constant for all pairs of oscillators. And what happens is that there is a nice way to represent this entire system by putting these particles on the unit circle. So the position of each particle is the phase. So as the particles oscillate, they will move around the circle. And what this diagram shows you is that as you increase the coupling constant, there is a tendency for the particles to synchronize. When they synchronize, they will move together, right? So what's the speed of the synchronized motion? So you can show that the speed of the synchronized motion is just the average of the natural frequencies. So if you take, for instance, in a simulation, this average to be 0, the particles will just stop when they synchronize. And where they stop, that depends on the initial conditions. The system is invariant and under rotation, so every time you do the experiment, they will stop at a different place. There is another important piece of information that you can take from here, which is a way to measure the degree of synchronization of the system. That can be done by computing the center of mass of the distribution of particles. You see, if they are uniformly distributed, the center of mass is going to be close to the origin. As they synchronize, the center of mass moves closer to the unit circle, so we have a big vector here. So the modulus of this vector is a good order parameter for synchronization. And you can calculate it by just averaging over the phases. You have this complex number with modulus and phase, or you can compute the vector itself. Okay, so one more slide on this. So if you now simulate this system with a finite number of particles, you measure the degree of synchronization as a function of the coupling constant. You see that at the beginning, you don't see much, and then eventually synchronization starts. So that suggests that there is a phase transition in the system, and Kuramoto actually showed that this is a continuous phase transition. He calculated the critical coupling constants, the shape of this curve, and so on. Okay, so first thing I'm going to show is how to generalize this to more variables, more dimensions. It's not my work. This is how I'm going to give you the reference in a moment. But to do that, we first realized that instead of representing each oscillator by the phase phi, we can represent them by a unit vector from the origin to the particle. So this unit vector is just cosine phi, sine phi. So if we know phi, we know sigma. But we can write down an equation for sigma. See how these vectors turn around in the circle. Since we know the equation for phi, we get the equation for sigma. And this is what we get. So the equation for sigma looks like this. So it's a vector equation. This W here is a matrix that contains the natural frequency of the oscillator, and this is how the coupling looks like in this representation. The order parameter, the center of mass, is just the average over these sigmas. So what's the advantage of writing the equation in this format? The advantage is now you can look at this sigma and say, well, these are unit vectors in what dimension? 2 dimensions, 3 dimensions, 4 dimensions. This can be unit vectors in any number of dimensions, as long as you, I think I have a slide for that. So this is, let me just give the credits. This is an idea by Edward Ott and collaborators. It's a paper from a few years ago. And the idea is that, since you are writing things in terms of vectors, you can think of these vectors to be in any number of dimensions. So they don't have to be vectors on the plane. They can be vectors in 3 dimensions, so you are in a sphere or in a 4 sphere and whatever you want. The only condition, the only requirement is that this matrix W has to be antisymmetric. Okay? So if you do that, you can guarantee that if you place the particles on the sphere, they will remain on the sphere. So the model is consistent in this way. And what is interesting about this is that if you do the calculation in 3 dimensions, you see that that phase transition that was continuous in 2 dimensions in the original Kuramoto model now becomes discontinuous. So the system synchronizes immediately for any positive value of the coupling constant, and it jumps exactly to 1/2. So there is a reason for that. You can calculate that. And in fact, they show that this phase transition is discontinuous in any odd dimension. So in 3, 5, 7, the transition is discontinuous, and in any number of even dimensions, the transition is continuous. Okay, so now I get to my work, which is a little extension on top of that. So the point is that if you wrote everything in terms of vectors, why keep the coupling constant a scalar? I mean, the natural operator on vectors are matrices. So can we extend the coupling constant to a matrix, and what does that mean? What do you get out of that? So it turns out that if you take the coupling constant that was here, turn it into a real D by D matrix and place it here and here, the system still stays on the sphere. For any real matrix, the system will stay on the sphere, and so it should work. So, but what's the meaning of that? So in order to understand what this coupling matrix means, I will first stay in 2 dimensions. So in 2 dimensions, you have a single phase, and this equation is identical to Kuramoto's equation if K were a scalar. So let's stay in 2 dimensions and take a rotation matrix as coupling matrix. What does that do? So if you work your way back from the vector equation to the equation for phi, you realize that what you get is this. So you get what is called the Kuramoto-Sakaguchi equation, where this alpha plays the role of a frustration. So it makes it harder for the oscillations to synchronize, because when the phases are the same, you still have some left over here that will change your velocity and take you out of synchrony. So, this is kind of nice, and it shows you how you can extend the Kuramoto-Sakaguchi model for higher dimensions, for instance. You just use rotational matrices. But what if you use a more general matrix? So what does a matrix do? So first of all, this is kind of obvious, right? What we are doing is that we are rotating every vector sigma j by alpha, right? So that's what you get here. The phase phi j is displaced by alpha. That's all you're doing. So, but if you have a matrix that is general, it's not a rotation, when you apply a matrix to a vector, you get another vector. And you can break this into a rotation plus a scaling. The only difference is that the amount of rotation and the amount of scaling will depend on the vector. Won't be the same for a vector as you have in the rotation matrix. So that's why we call this a generalized frustration. So again, in 2 dimensions, in order to see the effect of this entire coupling, you can take your matrix to be a sum of an antisymmetric part plus a symmetric part that we parameterize in this way. So it's a rotation plus a symmetric matrix. And if you work your way back into the angles, this is the equation that you get. And there appears this strange sum here of the angles. You have phi j plus phi I. So you say, there is no way this is going to synchronize. I'm going to lose my synchronization, because if the phi's are the same, you should get 0 change in the velocity, but here you always get something. But that's wrong. So in 2 dimensions, you can actually analyze the system, and this is the type of phase diagram that you have. So this is so these 2 parameters, j and k, are the amplitude of the rotation and the amplitude of the symmetric part. and you have these phases alpha and beta. So what you get in this plane is this diagram. These two lines that you see here, the slope is proportional to alpha, so alpha is represented here by this slope. If you are at j equals 0 at this line, you have the Kuramoto-Sakaguchi model, you have k and alpha. If you are at these lines, you can show that at these lines, the eigenvalues of the matrix are identical. So the matrix is proportional to the identity, and you have the Kuramoto model. Above in this region here, you get synchronization. But it's different from the Kuramoto model because remember in the Kuramoto model, when the system synchronizes, it can stop at anywhere with any phase. It's a random phase. Here, the synchronized cluster will stop at a very definite place, which is given by the eigenvector of the matrix with the leading eigenvalue. So that's interesting because if you change the coupling matrix, you can change the position of the synchronized cluster. You can control the synchronized cluster if you can control this interaction matrix. And finally, in this region here, you don't have stationary states. So the cluster of synchronized particles will rotate on the circle, and as it rotates, it will spread and contract again. The order parameter will oscillate. So this is a state that we call an active state, which is interesting and you don't see in the original model. Okay, so what if you go to higher dimensions? So in higher dimensions, you can actually find exact solutions for the model if you kill the matrix W. So if all oscillators are identical and they have 0 natural frequencies, you can show the two following results. So you see, the only thing that happens here is that you have this coupling matrix. So the eigenvalues and eigenvectors of the coupling matrix determine everything. So if the dominant eigenvalue, the eigenvalue with the largest real part, is real and positive, the stable solution is to place all your oscillators in the direction of these eigenvectors. So you take your higher dimensional sphere, find the direction of this eigenvector, you place your oscillators there, that's the equilibrium solution. And if you start anywhere else, they will converge to that position. However, if the dominant eigenvalue of k is complex, then you have ku is lambda u, that's the eigenvalue equation. Because k is real, u star is also an eigenvector with eigenvalue lambda star. And if you decompose the vector u into real and imaginary parts, do the same for the eigenvalue, you can show that the stable solution is a periodic solution that rotates in the plane generated by V1 and V2. So these coefficients alpha and beta can be calculated. They depend, so the period is the imaginary part, and they also depend on the scalar product between V1 and V2, which do not have to be orthogonal. When they are orthogonal, you tend to have no oscillations in the order parameter. If they are not orthogonal, you tend to have these oscillations in the order parameter. So if you put back the natural frequencies, then things become much harder, but we did some simulations, and I would like to show you what happens in 3 and 4 dimensions. Okay, in 3 dimensions, so this is an example of a coupling matrix, which shows a very simple matrix which has a rotation here and a single B value here, so that we know all the eigenvalues and eigenvectors very easily. This is the matrix of natural frequencies, an antisymmetric matrix with 3 independent entries. So we chose for these entries random values distributed as a Gaussian. So the center of this Gaussian is at omega 0, and the width of the Gaussian is delta. So we know what happens when omega 0 is 0 and delta is 0. That's the exact result. So when we change omega 0 and delta, depending on which eigenvector is the dominant, if it's the rotation or the real eigenvalue, you get these phase diagrams where you see here the rotation is dominant, so you get a small region of rotation close to the origin, but it changes into a static state as you move omega 0, or into an active state depending on which way you do. And similarly for the other part. So why is it doing that? Because the coupling matrix does not commute with the natural frequency matrix, so you have all kinds of crazy things happening. And in 4 dimensions, you can do the same, and you get this crazy diagrams. So this is numerical. We don't have these boundaries calculated, but you see that depending on the distribution of natural frequencies, you can move from one state to the other. So, so this is where we are right now. We are doing applications of this on modular networks, where we can choose different coupling matrix for different modules of the network and see how they interact as you couple them together. Next steps is to, yeah, this is an old version of the talk, yeah. Next steps are to add spatial dimensions into this work to do something similar to the swarmulators model, where you can also get spatial degrees of freedom. Extend this, this is the important part. We can also add 3-body and 4-body interactions to the model, which is something that people have been doing with the Kuramoto model. It can also be done here, and this has an effect on the synchronization that we want to check. So I think this is all I had to say. Thank you. Okay.
Okay, thank you, Marcos. I have a quick question for you. In equilibrium statistical mechanics, you have this concept of an upper critical dimension above which mean field predictions are quantitatively accurate. And here you have something like a mean field model where everybody interacts with everybody. And do you have the same thing? Do you have an upper critical dimension for this Kuramoto model?
No. In fact, what you have is, what matters here is if the dimension is even or odd. Yeah. And this has to do, this has a very simple explanation.
But this is the dimension of the order parameter, right? Like I'm talking about the spatial dimension where the oscillators are. That's one of your extensions that you're planning, right?
Well, yeah. But here I only have these internal degrees of freedom. So the particles are moving on this sphere. So there is no real space.
I got it. And this is like a mean field model, right?
Right.
And when you put it in a physical space. And do you expect to have an upper critical dimension for the physical space where these predictions start to?
I don't know. I don't know.
Okay.
Yeah, I don't know.
Okay. More questions?
So here, you do not have a dynamical noise on your equation, right?
No, yeah.
So you do not get like metastable states?
No, it's completely deterministic, yeah.
Because since you have a lot of dimensions, maybe you would get like the system stuck in some...
Yeah, you have to be careful here because you want your particles to remain on the sphere. And this is a vector equation, so the vectors can be anywhere. So what this equation has this property that if you start the particles on the sphere, they will remain on the sphere. So the vector field is always tangent to the sphere. So depending on what term you add here, you might break that, and the particles go away. So that's not what you want, because you want to describe phases that are oscillatory.
And if you add randomness to the matrix K itself, do you remain on the surface?
So this works for any matrix. Okay. And you can make the matrix depending on both I and J. You can have a matrix for each pair of interactions. That still works. Yeah, so you can have a distribution of couplings. So this is the simplest case of a constant matrix for every coupling.
And if it's a random matrix, do you still keep obtaining synchronization? Or?
Well, you see, in this case, if the oscillators are identical, then these results are very general. They only depend on the eigen. Well, if you have a distribution, I don't know. Okay. Yeah, then I don't know. Yeah. Okay, yeah.
more questions? If not, let's thank Professor Marcos Aguiar. And our last speaker of this session is Professor José Onuchic from Rice. I think there is people in the Zoom session.
Not enough hands. Not enough hands.
For the recording as well, sorry.
That's fine. Okay, let's start and see if we can do better, if we can put on present. Oh, yes, that's better. Okay, so I will talk a little bit about chromatin folding. It's an area we are very excited these days. When I run out of time, just tell me I stop wherever I am, because I think I have more slides than I should have here, but this is a very nice collaboration. Our Center for Theoretical Biological Physics involves some other senior collaborators in theory. We have a great experimental partner, that's Aries Lieberman-Eiden at Baylor College of Medicine, just across the street in Houston, doing the experiments. And I'm going to tell a little bit about it because it's sort of a cool field. Basically, if you look at the entire chromatin, and base, that means your DNA on your cell, if you're a eukaryote, is not just DNA. It's DNA with a bunch of proteins forming nucleosomes, forming structures. So it's a very complex system that you're dealing with. That's why I call chromatin. And these are the things that makes the difference. Just as a curiosity, I want to point out that basically every cell has the same DNA. Therefore, the epigenetics, what changed different cells, is just epigenetics. It's changing on this sort of not on the genetic that goes around, and so we're going to talk about structures of different chromosomes that humans. We are talking about this change into the objects. The other thing is, this is some result that people had about the chromosomes into the nucleus. That's a very interesting story because they noticed that when you look at these things at the interface, they tend to completely phase separate, and they form what people call territories. So these chromosomes get on mixture. And when they're mixture, there are lots of disease that come with it, so it's not a good idea, and how you do it. More than that, if you think about your entire DNA is about 2 meters long. It has to fold into a nucleus that's a few micrometers, and it has to avoid to make knots. So it's an amazing system that basically, if you really think about it, that can achieve those things. Is basically, as you'll see, if you put anything with a cable in your pocket, you'll see how many knots get formed. You think about how this thing gets the entire amount of collapse and can form that down. There's an experiment, mini experiment that came here, but there is this cross-link experiments. That's how people figure out how, on the old days, how to try to learn about the structure of these systems. And what you do is, if two parts of your DNA come together, you try to cross-link them, but form aldehyde or other chemicals that happen these days. Then you cut your system, you ligate, you put the biotin in the end, and then you try to get these pieces that come just the place they come together. And now you sequence these small pieces, and you see if you get some blue and orange together, it tells you that these two parts of the chromatin that are very far from each other, they are close to each other. As you can see, if you look at this experiment, there's nothing can go wrong here, basically, the amount of chemistry. So it's just amazing that this thing works. But people have been working for a long time, and they can do it. Just to put a few pieces of information as we go around, you get this contact map. Most of the information I tell you is in a resolution where each bit is about 50 kilobases, right? So think about it, just to have an idea. That's a little bit larger than a gene. A typical gene is going to be between 500 base to 5 kilobases, right? So basically, is in a larger resolution. Actually, it's very interesting that there's lots of information on chromosomes is on the size of 50 or 10 kilobases or coming down to nucleosomes. That's about 150 bases or or the 100 base, but in between, on gene size, we actually have very little information these days. You're moving there now. Also, these samples, when they do these experiments, they have about 1 million cells there, so you're really looking at the statistics of this context over 1 million cells. You're not looking at a single cell, and you can do simple calculations to know that basically if you had a single, people try to do what they call single cell high C, but you have very few cross links. It's a very complicated experiment that can give very few results. We actually did a model that comes, how do you come from this two-dimensional structure that these experiments give to you and try to get a structure. So our first type is to show that basically we did a polymer model for these things that people learn from these maps. When people came from these initial maps and they looked at their patterns of formation, they figure out most of these patterns are separated on what they call types and subtypes. We are called A's and B's. For people to understand a little bit about chromosomes, that's euchromatin or heterochromatin. Heterochromatin tend to be much more rigid, much more compact. Euchromatin tend to be less rigid, more expanded, and that's more most of the active genes are. Right? And these things tend to phase separate, and I'll show you a little bit about it, and people observe that from the experiment. I'm going to give you a little more information about it. But we have a model that if we learn these types, and actually can go to subtypes when people do these things that tend to phase separate. From this polymer model, what's the three-dimensional structure of an assembly you get? Chromatin, different than proteins, is very fluid. Actually, the dynamics is very viscoelastic, so you cannot talk about single structures. Actually, lots of mistakes when people just try to thread a structure into a single contact map, because it is really an ensemble of structures. But actually, I'm showing today is we just do these experiments to compare our results. So what we did, we figured out that actually these types, A's and B's, they actually relate to epigenetics. People have learned they're correlated to epigenetics. So we have a machine learning term that actually learn from ChIP-seq that tells you what's the information into these DNAs, which parts get methylated, acetylated, and things like that. So I have a machine learning term that learns the types and learn these three structures. Now we can compare now what's the equivalent contact map that comes through the ensemble structures. You can see what's the power for a method going together. Okay? So this is the potentials. I'm just not going to give enough time to give you details, but it's a structure we have done over the last 10 years since we moved to Houston to build this entire framework that's out there for the system to work. What's very cool is this potential is actually all these terms is built with a homopolymer term. It's actually a soft homopolymer, because remember that you have top isomerization into DNA, you can cut it, so these things can go across. Typical experimental data, you can do these things with about a 4 kT barrier for the soft homopolymer. You have a term, like I told you, that put this phase separation of these different types and subtypes. But you have an addition, loops are not too important at this level. When you go to lower resolution import, but you have really a term that's based on what you call the ideal chromosome, that instead of being interaction in space, they're interaction in sequence. So it tells you things that tend to be more close in sequence, they tend to form strong interactions. That's like in proteins. You have II+4 for secondary structure. It's a similar term for that. So the origin of this term actually, and then, as I told you guys, basically, on the first thing we did, we got one chromosome, that's chromosome 10, from a particular cell line, that's GM1278, it's one of the most studied cells and most stable, that's why people did it. And we can train these parameters using typical trick that you heard a lot about here on many other different problems, right? Going through this information, try to learn these parameters. And you just iterate and you get these parameters and you have a potential. After they are learned for this chromosome, we don't change them anymore. Okay, they're learning a single chromosome in a single cell line. We're going to apply to all the other chromosomes, the other cell lines, and it actually works quite well. So it's a very robust model that comes out of the system. Here's just to remind what I told you, that basically, as you look at these patterns, this is a piece of that high C map in the end, and I just want to show here that if you look at patterns like these different patterns of interaction, they actually get related to different levels of epigenetics, different levels of methylation, acetylation into the system. So that's why we can now learn these things. The other thing here is a comparison of a particular area of two cells GM1278, I told you, is a lymphoblastoid, it's a blood cell. This is a pulmonary cell. And I'm just to show you here that they have different patterns, and they also, clearly enough, they're going to have different levels of epigenetics. So with all that, that's how we can actually move forward and do into these systems. And just one more part, I want to point out that this term here that I told you about the ideal chromosome term, these terms tend to form helices. That's why when you look at the structure of this chromosome, they form helices of helices. They have sort of liquid crystalline nature into the system. And it's one of the reasons that actually avoid knot formation, right? Basically, sort of, if you have more local contacts, force yourself, compensate long-range contacts helps, is one of the terms that help you. But you can see this structure form that. And this tendance of this term really increases as you go to the mitotic chromosome that becomes very, very helical and very compact. Okay? This term here, also we treat as a potential also, is not really a potential. In practice, this comes from motorization. There are all these motors, what you call cohesion, condensing. They are the things that tend to pack the system. Condensing tells you something that to condense it, right? Comes with the name. But this works at the level much smaller than 50 kilobase. So in a sense, we treat that as an effective potential at that temperature. We create a temperature that tries to mimic the experimental motion of the chromosomes, right? But it's really, the process is active at a smaller scale, although you're treating it with an effective potential here. So just to put people to be on the same page. The second part we're going to try to do is this term here, this machine learning term here. And we have been working on that. You heard a lot about, from everybody here, about covariance. And I want to point out that basically what we do here is, the following. You look at all these chromosomes, okay? And now I'm going to break each chromosome as a collection of chains for each locus of it. The first locus on my chain here, on the sequence here, you could call like a sequence, a protein, was like a protein sequence, really a sequence. The first one tells what's the kind, these A's or B's, or A1, A2, all these subtypes. And the ones below is all this information I get from ChIP-seq data. How much each particular methylation, each particular histone is being methylated. So this is all ChIP-seq information. So I have all these sequences. And now I can look how you get types are related to this experiment, but we do covariance between these chains. That means we don't get just the relationship between the type and the experiment, but I also have the covariance between the experiments. So I get much more information. Now I think about it, we did a lot of work of these all proteins. We don't have time here on 20 minutes to go over, but these problems, like everybody here knows, gets fit back into a Potts model. Okay, so we have all the statistical fees that we need to do. We can just go there and copy, okay, and run the simulation and let the system to learn. And what you have now is a situation where you can actually learn these types and subtypes direct from the ChIP-seq information. So right now, there's something what they call the ENCODE database. And we are crazy enough to actually get the ChIP-seq information for all the cells they have in the ENCODE database. We create all possible 3D structures for them that people don't have. We compare to the ones that have high C, and they do quite well. But the three-dimensional structure is starting to come out. So it would be lovely to see if what we have been proposed, the kind of ensembles they have, is what agrees with experiments as more imaging come around. Right? So that's one of the great things I think to work on this field, because I think this chromatin field is the same stage that protein folding was when I was an assistant professor, where we had very little experimental information, and we could do models and predictions, and then the experiments start to come out. This is a field that all the imaging, not only high C, but imaging experiments are coming out, so can see how powerful this theory is at predicting all the structure. So from this figure here, it's nice to say that these are the experimental subtypes that the experiment tell me. This is the one we predict experimentally, and you can see it's reasonably good. And this is just to show some experimental results. We showed that basically, in principle, I don't have to include all the ChIP-seq information. I just have to include enough to be able to distinguish between these types and subtypes. So you're trying to figure out what's the minimal set that we have, because there's some experiments that people don't have all the information. So it's been a big game coming together. So what we are doing now is actually, here's some cases just to show you how powerful the method is getting. And it's very interesting because you start to see that basically a lot of the errors comes at this interface as expected, because they're making this discretization on these A and B types. That's a little bit arbitrary, right? But actually you do quite well on the predictions. And more interesting, now we can start from chip-seq information. Here, 2 particular chromosomes. We can predict the chromatin types, and then we can use the polar model to predict the 3-dimensional structures. So that's the power of the method. Now, if I get this ensemble of 3-dimensional structures, and we have a great software running for these things now, AMD gave enough computers for us that everything is running on GPUs, extremely fast. If you go to our website, if your run is not too large, we do that for you. Right? So basically, it's lots of groups are getting this structure. Like I said, I have all these things on encode. But the nice thing now, as I told you guys, is that basically, we can now get this ensemble of 3-dimensional structures that we predict, and we can say, do they at least agree with these high C maps? You have to be very careful in this comparison, because here is what you get from experiments, from theory, from simulations and theory. Here's what you get from experiments. And you don't want to only compare around the diagonal, because these always have a very strong diagonal, like I said, because of these local interactions that comes from these terms. But actually you want to compare far from the diagonal, and these are comparisons, and you can see these are not precise comparisons. The experiments are not precise. Now, what the experimentalists don't like, and we wrote in a paper, they almost killed us, is that actually the experiments are not perfect. The base high C experiments are not perfect. So we saw that this difference we're getting here is almost the difference that you get if you do 2 different experiments on 2 different labs doing the same cell line. So that's the precision of the entire field, both theoretical and experimental these days. But you can see the patterns are very similar. If you look at here, that means the 3-dimensional structure changes very little. Actually, one of the secrets that come from these things on the structures work for a long time, Bill is here and people know, is the following. We learned that from proteins, but applies here also. In proteins, if you get a few long tertiary contacts correct, you almost predict the structure, because you have that polymer. So it's very hard there's not too many ways you can put those things together in a 3-dimensional structure. So that means, what we learned from proteins, that basically, if you get about 60% of the contacts correct, and the others one are just made in random, wrong, there's no problem, you get a structure. Because when the system tries to say how you maximize the number of correct contacts, you actually get a structure. So the fact you have a polymer, these models do better. The structures you get when you start to compare to experiments, I'm going to show a little bit of imaging, do better than you expect, because of the constraint that you're not have a bunch of unconnected balls. They are connected by a polymer. So like I told you guys, we have as we have done in life for a long time, we have an entire website with all these models, the same thing we have for our protein structure predictions and simulations model. It's all available for everybody. You have to run. People can check and use your runs. And I noticed that's very useful to have other groups running. When I was in Santa Barbara, sometime Bill was there. Bob Scharifer used to run there. And one night he told me one of the great things I heard in life. He said, be very grateful to your scientific enemies. Nobody else will check every work you do for free. So it's very important to put your stuff there and get people check your code. If they find a mistake, they tell you. And it's good, particularly with code, as I tell the students, there's always a bug. Just keep looking, there is a bug. Right? So you can really improve by having the community using and working with you. I want to skip a little bit, a few topics, but I only have 5 minutes here. And it's just to show that now you can apply for many different cell lines. Here's just a few of them. We can go around and compare. But one of the most interesting things is that people are now doing imaging. And when you do imaging here, is like, these are experiments done in a group by Shuai-Zong, and she just got a particular segment at that time inside of chromatin. And she got very annoyed when she called me and said, you know, we repeat these experiments many times and we all never get the same answer. I say, I have told you it's an ensemble, and you guys believe you're getting a structure. So basically, the nice thing is for this species, we can now compare the segments, and I'm running out of time. Here's just two particular segments. One is that has no structure. You just get things more or less right in terms of regeneration and how these things go. And the other one looks like a dumbbell that's open and closed. And we're very curious about it, that you have these two minimas, because results do quite well. We can compare free energy profiles, everything just running over there. But you have this dumbbell, you have open and closed. But the cool thing is, in this dumbbell, most of the genes, it's a very gene area, but the genes are all on the link. So it's the first time that you have this kindergarten version of a functional aspect that you know that this particular chromosome can open and close, depend if you want to sort of expose all these genes or not. That's far from saying that we have understand function. This is just a simple case, and it's an observation. It's not like a theory or prediction, it's just more than observation. But it's an interesting result for the segments here that we have been doing these things. Here's just to show that if you mutate the sequence for residues that tend to be active, non-active, the active ones tend to come to the surface because they want to be on the surface of the chromosomes in order to do that. Finally, just to close, and I'm completely out of time, I want to say that one thing we're doing now with AIR is what we call the DNA zoo, where we're looking at the structures of the cells in all these different systems, all these different animals. And what we show is that basically they don't have all the structure. The idea that you have territories formation actually works for humans and for most highly developed vertebrates. But if you go to flies and mushrooms, they tend to have, instead of the chromosome separate, they have the centromeres or the telomeres of the chromosome tend to pack together. So as you move on these different species, you can start to observe these different situations. People can get territory formations you have, but you can have stuff like, for example, centromeres coming together or telomeres coming together, what they call polarized X. They have this different sort of situation. And just to close, I want to point that basically you can start to observe that when this state dominates, you get the chromosome separated like that. When this other state dominates, you have the centromeres coming together. And what you observed by doing a simple model here, that depends on the level of motorization. Right? Depends on how much. If you have a strong level of motorization, lots of condensing, you tend to form territories. I don't have time to go into details, but just so that's sort of very cool that you start to observe that basically, if you run a simulation with this model, as people observe on the experiment, you have weak territories and the centromeres tend to pack together. If you increase the compactation, increase this motorization, you start to create territory formation and doing that stuff. Then, what's cool is that at that moment, we start talking to Ben Holland in the Cancer Institute in Holland, and he observed, giving a talk, said, why don't you talk on that? And we started to work together. Ben noticed that he could, for example, get a human cell and just reduce the amount of condensate. There are lots of degrading, that's the way of doing. And you observe when you reduce condensate, you go from these states where you have territory formation, see all these different chromosomes are different, when you have centromeres start to get clusters. So as we said on the big propaganda of that story, you're able to convert a human into a mosquito, right? So basically it's a big transformation of the system going around. It's sort of a cool story that comes in there. I'm not going to tell you that, but sort of just stop here with this picture that tells you that you have many of these systems and they can pack together. So the entire story here is a very nice prediction story. It's really a way of doing good polymer models with some information theory that people like to do it, machine learning, learning these types. You can actually understand about the structure of these systems. It's interesting that now this mechanism we have been proposing that basically things are governed by this kind of potential where you have phase separation from different contexts, different bits, but you also have this sort of sequence term that we call the ideal chromosome is important, and it's a great physical problem. Is that enough? How far is it? I believe that's enough. These models have been good enough. We are now switching to many different other species. There are lots of interesting questions that tells you, okay, you separate into some 7 types and subtypes at 50 kilobase. Is there something essential about that? What happens when I go to a 1 kilobase resolution or 500 base resolution? We still have 5 types, or do I'm going to have to change the game, right? Basically, how that will relate. So there are lots of interesting questions, but I think it's a field that's really prone for physicists these days, and I think I talked for long enough. Thank you.
Jose, you contrasted proteins and chromatin. So I can think of a protein, it's a polymer, and there are contacts among different pieces of the polymer, and there are enough of them that are at sufficiently long distance along the chain that by the time I'm done, I have an essentially unique structure. If I imagine more generally, I have some density of contacts, is the transition to, I mean, it seems like in the limit that the polymer is very long, that there should be a phase transition to a unique structure. Where is that the sol-gel transition for the chromatin?
I don't think it's a sol-gel transition, but let me tell you what I think the transition is. That's an excellent question, because you don't have a single question. So what happens into first approximation, I don't have time to go into detail. These bit terms tend to be more attractive to each other than the A's. So the first approximation, if I want to do a very simple model, you could say the B's tries to get together, and when the B's try to form the score of B's, the A's form balls on the surface, and these balls on the surface, if you look at these different structures, they tend to bubble around, get together, and form. So you tend to form the gene expression bubbles to the surface, and these clusters are much more dynamic than the B cluster. B clusters tend to form big clusters that tend to survive for a long time. Here, there's a lot of dynamics. So the interesting question comes, I don't know if I want to call sol-gel, but it's really a transition, but it's a much less solid transition that you have on these interactions, in the sense that if you look, for example, that's interesting if you look for names. If I look on short times, and short times what I mean is, let's talk about the cell cycle. I'm looking at time about hundreds of the cell cycle. Then the system is really vibrating. It really looks like a very rigid system, right? But if I go to times of 10% or 20% of the cell cycle, then you see this more viscous dynamic. That's why it has this viscoelastic behavior. How they get connected to function is very interesting. Now, I don't think it's sol-gel, but it is a transition, but it's not as hard as proteins. That's for sure. But I think it's a good point. Yes?
Hi, Nelson. Thanks for the talk. The question is somehow in the same line as he asked. So I was worried about dynamics, and you just said a few comments about it. And I was wondering whether this kind of chromatin, the structure, this phase transition, could involve some sort of liquid-liquid phase separation, forming this like the nucleolus, like the membrane-less organelles within the nucleus. Have you thought about it? I mean, this is a hot topic now.
Okay, so first of all, I don't like the word liquid phase separation, because I think it's much more gas to solid phase separation, but it's a discussion we can have about surface tension here. It could be. In my model, I'm very agnostic about it, right? Basically, you could say that when I run my potential, I say there is this term, the phase separate. There is this term, but I didn't give you a physical potential. I'm talking on bits on 50 kilobase in size, and part of it's going to be that kind of situation. There's no question that chromatin has a little bit of it. But I think you're going to have a lot of these sort of transitions in every order, on membranes, on what they call these membrane-less organelles. This is going to appear. So the real physical origin, I think we don't have it. How exactly methylating a site to switch from an A to a B range is and it's not a single methylation, because remember, my beads are 50 kilobase. So the question of how methylate that site. It's the density of methylation on that site compared to the other thing, because you're having 50 kilobases per bead, right? So in a sense, that's what you have. So could be these transitions, basically. The scale that people have observed experimentally is much smaller. People talk about transitions on that size, on a lot of speculations on theoretical papers these days, but there's very little experiments to show that what's the physical mechanism at that scale, right? My son just worked on smaller RNA and protein things, phase separate on his thesis, And they tend to be much smaller clusters, right, than the size of the system. These systems, each bit here is about 0.2 micrometers, right? Basically, I didn't put all those scales. So something like that's taking place here. But how that scales on that scale, the physical origin of it's a very good question. I think there is a mechanism like that, because that's a mechanism that you have, and these things are controlled by these histones. You mutate more these histones, but how exactly this is charge control, or this is, and you can have basic liquid phase separation, this data people call by any possible mechanism.
But no one knows anything about it in any kind of system, how they control it.
No, I think on the RNA and protein system, people tend to go based. When people just do RNA with IPAs, they do quite well. And they can really control forming different kinds of bubbles and stuff as you move along.
We have time for one more question.
Is in your, what do you call, biological periodic table that you're trying to design in this picture, do you think that inside the simple model that's just polymer type of model, you are going to understand the basic mechanism behind to really design the periodic table?
Okay, that's what we hope. Right now we're in the period of observing these things, but there are a few things we have observed. I was going to tell on the time. You observe that when you start to have a system that's more centromere control than have territory formation, they tend to be much more sensitive to force. Okay, so cells tend to move more into that direction if you want to have cells. So the question is why some ones want to be sensitive to force or not. These are very interesting questions. All these things are appearing now. You have to understand that these structures are just coming now. There's very little dynamical experiments. Most of the stuff is just structure information. These are but I think it's a beautiful time. It's the time, like I said, it was like we were on protein fold in the 90s. You're making guesses, then the experiments are getting better, so it's going to become just an interesting field. And the beauty about theory is that you make all these proposals and let the experiments come, and the interesting part is success and failures are going to be equally interesting. Right? So basically, I think it's going to be a great time coming along it.
Okay, one quick question.
Quick question. Thanks, Jose. Essentially, if you look the same chromosome for different lineages, different types of tissues, you modulate the A's and B's that satisfy your model. How does it change your ensemble of your contact map, essentially?
Okay.
What's the idea? How do you explain the difference?
I show a little piece here just for a taste to show three different cell types. Three different cell types that I show you, I think, down here, where we show that basically pieces of chromosome that are inside move outside. Right here you have these cell lines, basically pulmonary cell, blood cell, and other cells, and you can see that basically as you mutate the kind of, if you do different epigenetics, here it didn't show different structures, just show that this particular piece that we notice was very different on that particular segment actually goes from inside to outside, because you have to become active as you move along. So probably there's some genes there. So what you observe, there are differences. Precisely how these differences are involved to function, I think we're just flirting with it right now, right? Basically, is how things get exposed and things don't get exposed. I could go through my entire speculation that basically most of these chromosomes, then in the end, you're going to have a model where they're coupled to the entire nucleus membrane. That's why on the case when you put laminin, an extra term that makes these things to bind to it, they bind the chromosome and some of the B's move there to bind. The center of them get exposed, and they can get the entire machinery of the cell into the cluster, so you try to have this A core in there. These are things you start to observe, and these things have to be exposed. But it's a very interesting question, and in terms of, and I could go through the list of things, we just don't have time here in 20 minutes, basically. Do the cells, as they move from metaphase, go the cell cycle, interphase, do they have enough time to terminalize? Or this system is actually off equilibrium? And the first guess we have now, and some experiments are in agreement, is that single chromosomes can actually terminalize in a cell cycle, but the rearrangement of all the chromosomes cannot. Some experiments show you have to go, just came out, they are right or wrong, but they claim you have to go through 8 or 9 cell cycles before you can actually thermalized. But remember, so it's a very interesting story because the system has to go from that structure to the mitotic structure and go back. So these are interesting things that come into this question.
Okay, let's thank again Professor Onuchic. Now we have a short break, 10 minutes, and we'll be back here at 5:05, say. Yep.
era o último que faltava.
Oh, I think we can resume.
Esse é o apontador é aqui. No meio. É esse aqui. Esse no meio, né? Para frente, para trás, a iluminação aqui não deixa.
Você tem costume de controlar o seu tempo? Não.
Eu vi você usando
Okay. I think we can start with the last part of the session with Giovani Vasconcelos.
Thank you. It's a great pleasure to be here, and I was here for the 5th anniversary, so I'm even happier now for I see that the child has grown, has become an adult, and it's so wonderful to be back in this wonderful institution. So this is another different talk. So I think Roberto said something to this effect, that the beauty of complex systems, that you see so many different systems that, but that's the interesting thing. So you get to know what other people are doing in different areas, but they somehow they have in some cases, a common strain at least. So it's the complex dynamics. So that's the focus of my talk today is in the complex dynamics. And I want to use turbulence as a platform, as you see, I would call it a universal model of fluctuation phenomena. And of course, this work of many collaborators is going back to a PhD thesis in, I think, 14 years ago or so by Domingo Salazar in Pernambuco, where I was. So here's an overview of what I plan to do, is to show you some recent work on this fluctuation phenomena. And I want to give this overview showing some recent papers from our group. And you cannot read from there, but they have 4 different systems here. This is fluid turbulence. This is a random fiber laser, so laser that emit intermittently, if you will. This is a integer quantum Hall transition problem, and you also see this fluctuation phenomena. And even in, of course, you are familiar, or some of you are familiar with fluctuation of asset prices in financial markets. So what's common about these very different physical systems, if you will, is that at least in our papers, they all share a common name, a common word. It's turbulence, 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 and turbulence. So I wanted to give you an idea of why this system can be put together. Now, I had this question mark there, in this universality class, if you will, of turbulent phenomena. So the prototypical or the typical turbulent phenomena is a velocity fluctuation in a turbulent flow. So here we have a laminar jet that becomes turbulent, and you see the formation of structure of different sizes. I'll come to that later when you try to model this in an effective way. But what you see when you measure, say, the velocity at a given point here is a function of time, and if you measure the increment of this velocity, the difference of successive velocity measurements, you see this fluctuation curve here. So it's pretty much like a random noise, and we'll come to that later. But this, of course, is well known, but you see the same phenomena in this random laser. This is the intensity of the laser that emits randomly, and you see fluctuation like this. And for the Hall conductance, you met the transverse or even the longitudinal conductance in a Hall strip, and you'll see fluctuation in this conductance as you vary the magnetic field. That's the fictitious time in our problem. But it's a similar curve to some extent. And of course, in price fluctuation, there's a foreign the exchange rate between several pairs of currencies, and you see the same sort of fluctuating curve. So what's common among them between this random picture is that when you measure the probability distribution of the increments, being velocity increments or price increments or intensity or whatever, you see first a heavy-tailed, non-Gaussian distribution. So that's in semi-log plot. So a Gaussian distribution here would be an inverted parabola, and you can see that concavity is upward. So quite different from Gaussian. And they look similar. That's very striking to me, that fluctuation in velocity has the same kind of probability distribution as fluctuation in prices. So my claim here, that's not a coincidence. And you can see also in other more complicated systems. These are quantum systems. They are more difficult to explain here, so I don't want to go into details about this lower part. But there must be some common dynamical fixture that gives rise to this distribution. So in the same way that we know why the Gaussian distribution is the normal distribution, it's because we have the central limit theorem. So here we don't have the same fundamental structure, but there must be something underlying the appearance of this distribution. And that's the question for today, to me, is try to explain how do this distribution come about, and why do they look similar? So of course, when I build a model and see that it's very reasonable that they should appear also in all the systems. And this model, we call it age theory. Age for hierarchical is a hierarchical formalism, if you will, to try to explain or to model the emergence of non-Gaussian heavy tail distribution. So it's based on very three general assumptions: local equilibrium, and then intermittence, and then a cascade model. So it's all this terminology, the concept borrowed from turbulence and actually applied, the same model that you apply to all these different systems. And so here's the main idea. The first assumption is that you have, of course, it's a multi-scale fluctuation phenomenon, so you have a structure of different sizes. And in particular, in the case of turbulence, now when you drive the system, your fluid, into the turbulent state, you inject energy, you form large structure, the large eddies. Now they split in smaller eddies and then into even smaller eddies and so on and so forth. So you have this cascade of energy from larger to smaller scales. And now when you do a measurement in a small cell here, or a small subsystem in a short time scale, you can assume that the system will try to relax to a quasi-equilibrium. So here you measure velocity at a short time interval. And if you were to measure the same, make the same measurement at a large time intervals, you would expect a Gaussian distribution. So you say that in the short time scale measurements, you also should obtain a quasi-equilibrium described by a Gaussian distribution. So the local distribution has the same structure, or is the same distribution as the largest scale distribution, but now you have a local parameter, this local parameter here, epsilon, that describes, your background variable that describes the local equilibrium. So it's like a local temperature. I'll come back to that. So the largest scale would be the reservoir temperature. So that's a local equilibrium, but of course this thing fluctuates. You have intermittent behavior. So in turbulence, this epsilon is the flow of energy going to the smaller scale. So it fluctuates in time and in space, even if you were to measure your probe. And then you have to take into account by assuming that the environment changes slowly, or at least lower than the velocity that you measure, the signal that you measure. And then when you acquire a large time series, now the distribution, you have to average the local distribution, your Gaussian, with the distribution, the probability distribution of your local equilibrium variable, the local background. So that's, some people call it, I think that's a mistake. I think I missed, well, let's go on. Now the second basic assumption is, or the third one actually, now I have to, let me go back, I have to measure, or I have to model how the local equilibrium, or the background variable that describes the local equilibrium varies in time. So it varies in time because energy is flowing from the larger scale into that smaller scale. So there was a cartoon here for this nested reservoir, but I think I missed it. So when I convert into PDF. So how do I model now the fluctuation of the background? So the main message that I wanted to anticipate that in our system, the physics is in the fluctuating background because the larger scale describes your quasi-equilibrium. So what happens in a turbulent flow? We have an energy cascade. As I said, energy is injected at the larger scale. It's dissipated at the smaller scale, the viscous scale. And then pretty much in this here, the so-called integral scale, you have a transfer of energy, pretty much conservative transfer here of energy from larger to smaller scale. So I wanted to model this. So epsilon, as I said, was the energy flow going to the scale that you measure, or one of those scales here. If you are somewhere here, then there's energy flowing from the larger, immediately scale above it into the scale that you are measuring. So you propose a stochastic equation for how the energy enters into this scale And that's essentially, I don't want to go into detail, but essentially a Newton's law of cooling, this first term, plus a noise term here. And you can explain why it has to be this, or there's another class of model I don't want to talk to. I don't have time to describe, but that's what you have. You have this cascade model, and your job is to compute the probability function of the level that you are measuring. And I think I missed another one here. There was another. So it'll be difficult here if I don't see. Yeah. So there was an important equation before. You can solve in some approximate way this set of coupled stochastic differential equation, and you can find the probability distributions of the Fokker-Planck equation for this smaller scale when you are making your measurement. So you can solve the Fokker-Planck equation for this variable, and here I had an equation for it, which is given in terms, an analytic solution given in terms of the G-Meyer function. So, but you have a solution, an analytic solution for the background now. And then if I insert that and do the average, and then this G-Meyer function, for those who know them, they have miracles, they perform miracles. And when you integrate a G function, this is a G function. If another G function, you get another G function. So in the end, you have your final distribution. That's called the signal distribution. For this case in particular, you can show that this function has power law behavior. So that's how power law, this heavy-tailed distribution arise from this composition of a distribution. And you can show that there's a second class that gives you a stretch exponential. I don't have time to go into the details, so let me show you some examples, and then we can discuss it later. So the first one is, again, going back to fluid turbulence. And now it's a bit more tricky because in actual experiments and in simulation DNS, you can, and there's some very fundamental results due to Kolmogorov that show the distribution is asymmetric. So you see there's a heavier tail here in the this delta V, so the loss increment, the negative part is heavier than the positive part. So you have to take into account, you have to break the symmetry, and then it becomes a little bit more complicated because your Gaussian has to have a non-zero mean. But you can do this, it's a bit more complicated, but you do get, I forgot to mention perhaps, but you do get an exact solution in terms of generalization of these G-Meyer functions. They are a special function, complicated function to some extent, but they're just special functions. And then what you do, then you fit into your model. There's one equation missing, so it'll be difficult to explain. But in this plot here on the left side, on the left-hand side, this is the histogram from the data. This is numerical data that we accessed. And then you compute the distribution of that variance. You can construct a series of the variance, and then you do a histogram of that auxiliary distribution, which is your variance. And then you do your fit in here, not in the signal, because I told you I had an explicit analytic solution for the variance as well. So you can do your fit in here, use the parameter, and you can show here that the fitting is good. There's one parameter you have to choose, is the number of level in your cascade. You don't know, so you have to try 1, 2, 3, and see which one fits best the data. In this case was 5. And then let me come to financial markets very briefly. Now it has been for a long time suggests that financial markets in particular and foreign exchange markets in particular are turbulent, and you can do the same sort of analysis. Now we have a cascade of information now. Now information cascades down from larger horizon inverters to medium-term to short-term inverters. So that's how you have a cascade of information that's reflecting the volatility Oh, that's our parameter now. And you can do the same kind of modeling. Sorry for the, and here there was an equation for the same. So now it's the volatility. See, when you convert from Mac into, see, I should have used mine. Yeah, I convert into PDF, and then usually when you try to do and insert into a Mac, Windows, hell breaks loose. Oh yeah. So no, next one, please. So you do the same thing, and then that's just the final result. We analyze the Euro USD. No, it's not going to happen. Yeah, so you can just leave it there without the presentation mode. Maybe you can, and then you see maybe I can just mention it. No, it's the previous one. I always skip. Oh, is that? And the plots, we had the plots before. Now it's all black, it's all white. It's black. Well, okay. I'll try to explain. We analyzed the 3 largest currency pairs from Forex, and we showed that the larger the market, the larger the number of scales in the information cascade, which is a very, very interesting result. You could not guess in principle. So it's like in turbulence, when you increase the integral scale, the integral size, like increase the Reynolds number, and then the number of scale in your energy cascade increase. So you see something similar. So it's the first actually concrete signature we call of this information cascade rather than just an analogy. And I think I will how much time do you have? So, yeah. Okay, so I want to now speculate a bit more now. I wanted to show you the formalism. I don't want to talk much about the integer Hall quantum transition, but if you just show this picture, that's what a trained eye like myself do. This is the density of state in a Hall strip as a function of the magnetic field that you apply. I don't want to go into the details, but if you think in a semi-classical way, this is like showing the path of the electron. It's one electron, so the Schrodinger's equation, tight binding approximation. And then that's like you cannot see from there, but looks like some eddies being formed. So that's a turbulent behavior in an electron motion in a disordered 2D medium. But that's all. The same you find in a random laser. So now it's the photons that are doing this making edges and breaking edges and so on and so forth. So there's always this structure multi-scale phenomenon going on. And finally, if I can just move, let me speculate that, see, I have 1 minute now within my, so that's good to use. It's good, not counting the interrupts. Now, there's one, that's a biophysics section. There was one. Can you go back just a second? So I lost a couple of slides, but let me try to explain in words what I wanted to say, because I knew it was a biophysics section as well. Some people claim, and I'm sure that they are right, that the brain is turbulent, right? At least my brain is quite turbulent. I don't know what the Reynolds number is, but I can assure that's not small, at least when I'm stressed out. But what they measure is from ECG, and they just measure the spectral density, and they see some power law, and they associate with the Kolmogorov's law, and they say, well, the brain is turbulent. Now, with this kind of approach, you can actually probe what the distribution is, whether there's really a cascade in this signal, how many levels do you have, how the turbulent state varies as you vary, depending on the state of your brain. So that's what this is slide that's missing. We're trying to get access to this data. It's difficult to get data from people from this field. But if you have the data, then you can actually test, rather than just making computing a scale exponent. You can actually test whether this idea that the brain is turbulent is actually, fits into the turbulent model, rather than just by reasoning by analogy. And then there are many other systems. You see this fluctuating phenomena. If you have multi-scale, then you are bound to get this non-Gaussian distribution in our model. I'm pretty sure you covered that. And I think that's all I have time for. Thank you.
Thank you. Questions?
So what's the number of levels of the brain?
We haven't the data, but you see, even in I can again reason by analogy. So in the largest data set that we use for the fluid turbulence, the DNS simulation with the largest Reynolds number they had, we get up to 7 levels. So, and actually in turbulence, you can actually estimate a priori if you have the larger scale and you know the smaller scale, and you say that they are halving, so the length divides by 2, and in each level of your energy cascade, you can actually estimate how many levels do you expect from the Reynolds number. So that you get 7. In other systems, in the financial market, the foreign exchange market, we get the largest market has n equals 5. So it has to be within that region. So even I don't think the brain has so large a Reynolds number that you give you n larger than 6 or 7. That's my guess.
In what I would call the real turbulence, like the one you just faced in the early this afternoon, we also find these levels, when you have weather type of turbulence. It's also is like this, or we have in this artificial turbulence, like the market, something that's having these levels.
Now, I was in 2 parts. In the real turbulence, in atmospheric turbulence, you do have this non-Gaussian distribution. That's a very characteristic of turbulence flow. So you see the same behavior there. We haven't actually modeled this data. We are collaborating with people from what's called the environments engineering department that have access to data from the Amazon forest. And then now we have the effect of the surface, above the trees. So that might change the distribution, but you still see something like that I showed you. So that will be found in any turbulent system, in fluid turbulence. Now, coming back to financial market, you see the important thing is not, you don't have a Navier-Stokes equation there. So the important thing is to have some local, somehow way to pass this information from one time scale, in that case, to a smaller time scale, and then to even a smaller time scale. So whenever you have this cascade, whatever the actual physical interaction is, you are bound to get something like this. So that's why you say that turbulence is like a universal model for this fluctuation phenomenon. Whenever you have multi-scale structure, that the smaller structure, the largest structures feed into the smaller ones, and so on and so forth. That gives origin to this non-Gaussian. If the flow was stationary, steady, then you get Gaussian distribution. So the intermittent flow between scales, that you lead to this non-Gaussian distribution, whatever the system is. That's our claim.
Okay, thank you again.
Thank you.
So next speaker is Adilton Carneiro.
Hi, everyone. First of all, I would like to say thank you for the organizer of this very nice symposium. I'm learning a lot. I feel as a fish out of water. I learn a lot because you can start, please. maybe my mission here is talk about what we are doing in our department. It's a department of physics, but when this department was created, it was focused in medicine applied, physics applied to medicine and biology. So you look for physics properties, physical techniques to create some diagnostic or therapy application. When you think about medical physics, you mean, oh, it's ionized physics. But it's maybe my mission here to say that what you are doing beyond ionized, what physical protocols, process, technique you have been using for diagnostic therapy. After my talk, I have a colleague, Professor Costa, maybe you complete with about biophysics and your group. But in our department, most of our professors are physics that work with instrumentation and theoretical applications for physics applied to medicine and biology. So anyway, what we're doing. First of all, I'd like to just say about what happened. I don't know. Yeah, we have a graduate program. It's about medical physics. The graduate program is physics applied to medicine and biology, as I told. It started almost 4 years ago. Undergraduate students in medical physics. But this student has a good connection with people in different area. I know that the focus is ionized because the hospital application, but most of them work with the research in our group, apply optical, magnetism, materials to look for a diagnostic and therapy. So as I told, most of our research is focused in physics applied to medicine and biology. Where you are, at Ribeirão Preto, campus of University of São Paulo, in the Department of Physics, as I told you. Everyone is invited to go there, please. Bring a good shop. Ribeirão is a very nice place, a factory of good beers. A good campus is look like a florist, but we are here. Let me see. Okay, in our department. Anyway, so our main research, biomedical instrumentation, ultrasound elastography, acoustography, tissue mimic phantom, magnetomotive. What is it? I don't know. This is you have two. Let me see. Okay, no problem. I don't know if you delete the first one. What I told you, I gave you today in the afternoon. No. Oh, please. Yeah. Because I remember that I deleted some slide and fixed some errors. I'm sorry. Yeah. You put here medical physics. Here, medical. Maybe I think it was this one. Medical. Yeah, exactly. Thank you so much. So, our motivation, look for protocol process to map viscoelasticity of the tissue, for example, for diagnosis and therapy. And create new ultrasound, mechanical waves technology, for example, investigate gastric problem using ionized system, molecular image, develop intelligent nanoparticles, create drug delivery protocols, therapy, ultrasound thermometry is a good point. Ultrasound thermometry. If you have time, you discuss about this. Develop a theranostic system. This is the main focus, theranostic system. When I say about theranostic system, I mean combine in the same system a diagnostic and therapy simultaneously. Because nowadays, if you have a cancer, you get a diagnostic. After you submit to a therapy, maybe 15 days after, 1 month after, depend of the opportunity line of in the hospital. So this is the idea to look for personalized nanomedicine. Okay, to do this, you look for main physical application. For example, mechanical waves. Okay, I can use ultrasound, but what you're doing here? I can apply a ultrasound pulse, high frequency, and this pulse have an interaction in the tissue. And I will look for low frequency. So I get a signature from the mechanical information because this ultrasound interaction, for example. My answer is low frequency. Anyway, what you can do this? Look like my body's a combination of a lot of small diapason. Because if I apply ultrasound pulse, it's a mechanical, it's like a touch, and it creates a sound. As you learn about in physical too, about this, use a diapason to different frequency. For example, this is a bone image use ultrasound. You never see an image use ultrasound like that, because ultrasound for soft tissue. If you talk with a doctor about, oh, I use ultrasound for bone, oh, it's crazy. It's not make sense. It's not work for bone. Okay, you don't have a clinical protocol, but you can do this using this protocol. There is a lot of equation, mathematical problem behind this. But anyway, I use a big sounds as transducer and ultrasound transducer to apply a focused beam. And after barbecue, you keep the piece of bone of a chicken, the pocket, and go to the lab and play, and you have an image like that. I'm sorry. I image look like equivalent to X-ray image by ultrasound. Another, it's like you can find the literature some results about this. Another problem is, you need to train the doctor, a surgery or a diagnostic. When I say train, not to create a phantom, create a mimic material, just a tissue for mechanical. But if I apply for MRI, for example, I need to learn about what magnetic properties of this tissue. If I use for ultrasound, I need to think what the mechanical properties of this tissue. So I need to create a material that's available, that's able to produce an image with using main protocols, in this case non-ionized, that can help the doctor to decide. And it's very important to train. For example, some surgery, brain surgery is new now. Most of brain surgeries, the doctor didn't have this kind of surgery before, need to train you. So you can create this simulate, this mimic material, for example, breast, anyway. Just as an example, I have here at Ribeirão Preto in the last 4 years, 2 cases. One was very famous, it's cranial twins that it separated the head, for example. And our lab needs to support our research because all part of the surgery was trained before. You create equivalent, the growth to train this doctor. But to do this, you prepare the material with magnetic ultrasound properties to create the image in the MRI and ultrasound system, for example. So use a lot of physics behind. It's just an example to simulate the skin anyway. Another point is now you more about nanomedicine, as I told you. Well, you combine ultrasound, magnetism, and light. How can do this? Okay, if you apply a magnetic field in a nanoparticle, you have a magnetization, have a subatometry. But if you apply a pulse or high frequency, this nanoparticle create a force between external field and magnetization of the nanoparticle. You vibrate this nanoparticle. I'm telling about nanoparticles. And I can get an image of this vibration. I can use ultrasound because ultrasound is a mechanical wave. I'm not telling about ultrasound from the hospital. I'm telling about ultrasound protocol, ultrasound physical technique. So you can apply ultrasound to detect the micro or nano vibration of this nanoparticle. Oh, good. If you have micro vibration, I can get information about the vascular elasticity of the tissue. I can create a molecular image, for example. Okay. Go there. So you apply a force, this nanoparticle vibrate, and you create this kind of a diagnostic. Anyway, you have here our protocol, all mathematical behind of this protocol, for example. And if you apply, as I told you, a wave or a pulse, you vibrate this nanoparticle. But you need another protocol. How I can get this information, create an image? So it's not easy to get, okay, just a signal, I know, the phenomenon I know, but how you can transfer this phenomenon to create an image protocol in the real time. Anyway, you applied a magnetic field and detect many images before and after magnetizing, for example, and you can fit this data anyway. You can apply protocol to look for any information, each piece of the map of the displacement, and you can create this kind of image here. It's a kind of seismic wave. If I throw away a stone on a swimming pool, you create a longitudinal wave. I can get a picture and can see the wave propagation. I can do the same inside of the tissue. Use ultrasound. Can you please, it's a video. Can you run, please? Yeah, thank you so much. So you can see, what is it? I have a seismic wave inside of the biological tissue. How can you create this? This biological tissue is marked with magnetic nanoparticles, and I apply a pulsed magnetic field. You create waves, spherical waves. That's I say seismic wave. You are able to detect this wave inside of the tissue, use ultrasound. Oh, good. If I change the water of the swimming pool, I say I can calculate the density of the water, for example. You can do the same. So you can detect this wave by ultrasound and apply some calculation and detect the viscoelasticity of the material, for example. You can detect the stiffness. Good. I know that you have a cancer. The main physical parameter that the doctor used until now is look for the stiffness. I can say by bionic teeth. Use combined ultrasound and magnetism. Okay? Good. And now it's with me. So I can mix it. As I told, nanoparticle, I can look for just whereas I can see this color image because I have nanoparticles, and here you don't have nanoparticles. Apply the same protocol, for example. So it's a good technique to localize magnetic nanoparticle distributed inside of a tissue, for example. Another point is inside of the gastric. I can use this nanoparticle to mark food, and I can get information about the viscosity of the element inside of the stomach, for example. You compare two techniques. It was a protocol. This transducer is a susceptometric and ultrasound together. It was a patent in our lab, and you can see fitting both the same results. You can see that if I use a viscous food, the mixed is faster than is like solid food, for example. Anyway, just you can localize the volume of the organ. Now I told about theranostic. And I'd like to combine all information, light, magnetism, and ultrasound. Just to repeat, theranostic is simultaneously diagnostic and therapy. If you need a thermal image, what do you use nowadays? What do you have in the literature? Infrared camera, no? You have an infrared camera, you have an image. What we are proposing, to use ultrasound to get image, because infrared, the image is surface on surface. So you need you don't know exactly what comes from that image. Okay, depends on the target. Pay attention. I have here an ultrasound system. Okay, just the information. This ultrasound system, I can use the same ultrasound in the clinical, but it's open for us. You can program this ultrasound. So it's a special one. Okay, it's not work with using the conventional one from the clinical, for example. It's a special one. You use all technological system because you can create an image, real-time image, but it's an open system for us. Good. I have ultrasound. I have a system for magnetic pulse. I can place the frequency. Low frequency, I vibrate. High frequency, I heating. It's the same system. Good. So I have a sample with the magnetic nanoparticle in the middle. Thank you. And you apply a magnetic field. You excite it mechanically. I get information about the displacement. So it's in the color. image here is displacement of the nanoparticle. Just whereas I have the nanoparticle, I have the displacement. So I have a viscoelastic information. Here's a beam modes are mechanical information. And what is it? It's a thermal image. Because if you are heating, the vibration change also. The amplitude and frequency you change. So you can create diagnostic applied therapy by hyperthermia and control the hyperthermia. Use the heat image, okay? So it's very interesting. Another point is neuromodulation. You learn that you can apply TMS, transcranial magnetic stimulation, or electrical stimulation. But it's possible to apply a mechanical stimulation. There is some interference on the brain, on the body. Pay attention, again, please. There is a video, please. Pay attention to this image. I'm sorry, again. You apply a ultrasound, a pulsed ultrasound, in a part of the brain, like for example this point. It's a small mouse. Please. Oh, let's again. Please, again. It started, I don't know what happened. Let me see. Oh. Now, okay, come on. Was running. Are you back? No, I'm sorry, it's here. Are you back? Okay, you can run after. Let's continue in this part. Another point is combine light and ultrasound, photoacoustic. But I can use the ultrasound system and create an image. Everyone knows that photoacoustic, if you apply a light, you absorb this light, you have a thermoelastic phenomenon, and it creates a wave. How good. If I have a mechanical wave, I can program my ultrasound to detect this wave. I just change the frequency. If I create a high frequency, I will be able to use the ultrasound to detect this mechanical wave created by photoacoustic phenomenon. Okay, so I can use an intelligent nanoparticle, as I told you, for drug delivery, as a contrast to target the tissue. Anyway, main application. I just you skip this part, as I told you already. So I can create an image like this. It's ultrasound transducer, apply light. If the tumor is more oxygenation or less oxygenation, that you can see some contrast because the oxygenation, you change the attenuation of the light, for example, so you can see the tumor. Anyway, it's very interesting. Now try to, okay, now it's okay. You can see, for example, the right leg moving. Because I apply Fox ultrasound. So when I apply Fox ultrasound, it's a neuromodulation, a neurostimulation, and the mouse is moving the right leg. Now you change a little, just have a small displacement about 2 millimeters. It's another part of the brain. And you can see now the left leg. Just a second. In this moment you are looking for, because it's very precise. No. Okay. So it's possible to use this kind of information to stimulate to diagnostic and therapy. Anyway, our main challenge for this diagnostic is these nanoparticles. Nanoparticle with contrast for magnetic, contrast for ultrasound, contrast for light, for stimulate and diagnostic information. This is some part of our lab is some protocol, some platform. You are using, as I told, main physical information to create this protocol, neuromagnetism, electromagnetism, ultrasound, light, everyone combined to create new diagnostic and therapy system. Okay? Anyways, our connection and thanks so much for this group. Without this guy, you don't know anything. Okay? Thank you so much.
Thank you. That's very interesting. I'm wondering about delivering the magnetic nanoparticle to the specific parts you want to study. How much of a challenge is this? Or
Deliver?
Yes, to put the magnetic
Yeah,
nanoparticle.
good question. Yeah. When the big challenge is the volume. For example, iron liver, inside of the liver, for example, is a natural iron come from or you receive like a transfusion, blood transfusion, for example, you deposit you store the iron, or you have like a hemochromatosis, for example. Anyway, the volume is so small, so you use magnetic technique like a bioseptometry. By ultrasound, you look for, increase our technology to look for exactly this kind of application. For a while, this force is so small, and not enough because a little depth, it's not enough to vibrate and detect by ultrasound. You need a high load of iron, for example. But it's possible, okay, in the high level. But using susceptometry or MRI is better. It's so sensitive.
More questions? Okay, so thank you again.
Thank you so much.
So we are ready for the next talk by Antonio Costa.
Okay, let's start by thanking the organizers for the invitation to be here today. It's a pleasure to be at the IFT for the first time and see some old friends. I believe that this afternoon has been a kind of a switch gear kind of talk all the time, right? So we're gonna switch gears again, right? So we'll go back to molecular biophysics, but not the theory as Nelson just showed us, more kind of experimental work. Can we just, yes. I have to thank Adilton for the introduction about Ribeirão Preto and our department. So our department, as he said, is mainly focused on applications of physics to medicine and biology. And I'll show you some of the things we've been doing in the group using physical methods to study macromolecules, biological macromolecules. And the thing I wanted to focus today, to emphasize, to show you, is how this thing called biomolecular condensates can be used to organize the intracellular milieu. So I don't know how many of you are familiar with the organization of the cell, but this is a high school textbook figure. So that's the way we learn when we are in high school. So our cell is divided by this membrane there. It's called the plasma membrane. And within it, we have several what that's called membrane-bound organelles. Probably the most famous one is the nucleus, okay? So we just heard from Zanelso about the things that happen inside the nucleus there, especially the nucleolus and our genetic material, but there are also other membrane-bound organelles within the cell. For example, the endoplasmic reticulum, the Golgi complex, and so on. So that's the traditional way we think the things are organized within the cell, and that gives us our cells the capacity of running a biochemical reaction in a very controlled manner, because you can then concentrate molecules in a determinate place of the cell, for example, inside the mitochondria, okay? So we can do better nowadays. We can actually look at pictures of those membrane-bound organelles. Those are some of them, okay? So the Golgi apparatus, the chloroplast, the endoplasmic reticulum, and the mitochondria. All right, we can even do a lot of physics, both theoretical and experimental physics, to understand how the components of these membranes function or how they organize, and that's a very rich and prolific field of investigation. We've been doing lots of work about that too in the lab, but that's not what I'm going to show you today. I want to show you what happens when within this very crowded intracellular milieu we put together molecules with a specific type of characteristic, okay? So we think that our intracellular space is something like that. That's kind of true for the in vitro experiment, so we can imagine that the molecules are like that, but our intracellular space is more like this. So the molecule concentration within the cell could be very, very high. But that's actually a video, but I will not bother with that, okay? So over 100 years ago, this guy A.B. Wilson came up with the idea or the suggestion that the cytoplasm of cells was not actually, let's say, a liquid in the way we think that water is. Okay, so that's a paper published in Science in 1899, and there he already kind of speculated that the living protoplasm, that's how they called the cytoplasm at the time, was actually was a liquid, but rather a mixture of liquids in the form of a fine emulsion. And that's the image that they published there at that time. So imagine that they had to look at the microscope and draw the whatever they would see in the microscope, because there were no such a thing like this, right, to take pictures of the microscope. So pretty much at the same time, this guy, T.H. Montgomery, also published a paper in saying that, well, not only the cytoplasm does not behave like a proper liquid, or but inside the nucleus, there are structures, that is puncta, that look like some sort of emulsion within the nucleus. That's called the nucleolus. That's where our ribosomes are synthesized, okay? So it took us, or it took the mankind or the scientific community more than 100 years to finally show that those type of arrangements, these emulsions of these liquid compartments within the liquid, really or indeed existed within the cell. So this was a paper published also, coincidentally also in Cell in 2009. So the idea of E.B. Wilson was published in 1899, right? So this is 2009 when Brangwyn and Hyman finally published data showing that within this the C. elegans germ line, okay, this P granules, this called P granules, which are involved in defining what will be the germ lines in this C. elegans organism, was actually a liquid separated from the liquid of the cytoplasm. So this happened through what is called, we all know about it, liquid-liquid phase separation. So liquid-liquid phase separation, I know now that Zenos doesn't like the term, but I'll just use it anyway. Yeah, I'm just teasing you, yeah. So, we know a lot about liquid-liquid phase separation because the material guys know a lot about it, okay? So this was the first time that this type of phase separation was detected within the cell. And the guy showed that, so that the P granules, you can see here in green, so they are labeled with some fluorescent markers, okay? They concentrate in one side of the germline, on one side of the embryo, okay? So in this half of the embryo will be the germline of the C. elegans, right? So they run a whole bunch of biochemical experiments to show how that happens, but what interests us right now for this afternoon is that they show that these P granules were actually like liquids. Let's see if I can see. So that's the P granules. They can just like drip, they coalesce, and they have this liquid-like behavior within the cell. So they showed that in 2009. All right, so after that, we had an explosion of people. Several groups in the world start looking for other examples of liquid droplets within the cell forming through this liquid-liquid phase separation. And this is an account of what we have found so far. And remember the nucleolus I told you about just a few slides ago? So 2 years after showing that the P granules were like liquid droplets within the cell, Brangwyn and Hyman also showed that the nucleolus, okay, the nucleus, this puncta within the nucleus, are also what is now called biocondensates formed through liquid-liquid phase separation, okay? So these are all the places, all the examples within the cell that we found, that we know now that are these liquid droplets doing something. So this is a very effervescent moment for the field because there are lots of open questions there. Probably if you ask me a question, I'll just say, we don't know. So what is this for? What does the cell use this for? So we're not really sure. There's no common theme still for what the cell does with this kind of organization. But it is there, okay? So how did we, this is just to show you how kind of state-of-the-art of the field. So this is an editorial published in Nature in 2018. So this is all new stuff, okay? So, and the guys are saying there that, well, this is a new phase, making the kind of a joke, in cell biology. So we have to consider that the cell can organize its intracellular space using not only membrane-bound organelles, but also what's called now membrane-less organelles. So how did we enter this ball game, our group? So we were there years ago, interested in a family of proteins that are Golgi resident. So they are located in this organelle, which curiously is a membrane-bound organelle, okay? So this is called the Golgi reassembly and stacking proteins. The acronym is GRASP, okay? So the Golgi apparatus, for those of you who are not familiar with it, it's one of the easiest organelles to recognize in the cell because it has this typical arrangement of, is a stack of cistern which are glued together, see these bridges there. So this family of proteins, the GRASPs, were firstly identified as the glues to bring together the stacks of the Golgi apparatus, okay? So the Golgi apparatus is like the post office of the cell. So they take newly synthesized proteins from the endoplasmic reticulum, they chemically modify them, and then they destine them to their final destination, either within the cell or to the outside world. So that's called the conventional protein secretion mechanism, okay? Curiously, this family of proteins, the GRASPs, more recently were also identified as being key players in what's called the unconventional protein secretion. Okay, so in the unconventional protein secretion, proteins that do not enter this conventional route are somehow deviated from the Golgi apparatus and reach the extracellular space anyhow. And these unconventional protein secretion mechanisms are usually triggered or they start when the cell is submitted to some sort of cellular stress. So they are really important when we are sick, when the cell is sick. Okay, in one of those mechanisms called the type 4 UPS there, the Golgi bypass. So this family of protein, the GRAS, they do some sort of schizophrenic kind of behavior. So they somehow leave the Golgi apparatus, they swim back to the endoplasmic reticulum, there they engage in some sort of carrier, and then they keep secreting things outside to the outside of the cell. So it's kind of a desperate measure used by the cell to keep working, to keep secreting molecules to the outside world. So we started, we are a molecular biophysics group there, experimentalists mainly. So we started studying this family of proteins, doing the regular things we do. If you guys are interested in details about the experiments there, we can discuss later on. We just don't have time to go through all the details. But at some point we found out that these proteins, these GRASPs, were actually not well-behaved globular proteins that we can treat as a ball. They work actually in the realm of intrinsically disordered proteins. So we have an expert or two experts here at least in the audience on IDPs, Zanelso and Vitor over there. So these proteins, IDPs, intrinsically disordered proteins, they are very good to do a lot of stuff in the cell or in your system because they are very flexible and very promiscuous, so they can interact with a whole bunch of partners. And that was not known at that time. So there's just some collection of results from our group, mainly spectroscopy there and some biochemical experiments as well. So we even dig deeper in this thing. So we separated the pieces of the structure of the protein, and we identified that this intrinsic disordered region was actually concentrated in one of the domains called this protein has mainly three domains. Two PDZs, which are interacting domains that govern interaction with other molecules, and one SPR domain that's completely disordered. Okay, we identified that this one of the PDZs there that was imagined or supposed to be well-ordered and well-behaved, it was not. It was this domain that was the intrinsic disorder one, okay? And that, showed us why this PDZ1 domain was actually the one interacting with a lot of partners in the cell and not the other one. And that's amazing because as we are talking in the coffee break, IDPs are very flexible, so dynamics play a very important role there. Right? So although the 3D structures look very similar of those two PDZ domains, it's the PDZ1 that interacts the most between the two. And that's because of the flexibility, because of dynamics, okay? So at that point, was nothing new there. We even found out that those things could form amyloid-like fibers, okay? either both in vitro and in vivo. So that's data we collected in cells, okay, in yeast. So this is the grasp from yeast. We are just showing there using this fluorescence lifetime imaging there. It's there's a fingerprint for the formation of amyloid-like fibers. We show that these proteins could form amyloid-like fibers within the cell. So that's the bright field image. So that's the yeast, one yeast cell, and you can see that when it colored with, in its inside, it's showing that the protein is forming this amyloid-like fiber. So at that point, we are kind of happy because we are finding out a lot of new information about these GRAS proteins, but then we came across with this paper. Okay, that's a 2019 paper published in Fab's Letter. That's a very traditional journal in the field, and written by Jim Hoffman. So for those of you who don't know Jim Hoffman, he's a Nobel Prize laureate in physiology and medicine, and curiously, he's a physicist, okay? His background is in physics, and then he moved to biochemist, and then cell biology, and got a Nobel Prize in physiology and medicine. And there he's saying, look, the Golgi stack, you see, is like a liquid crystal, phase-separated liquid crystal. And we were not aware at that time about this liquid phase separation and condensates. And immediately when we came across this paper, we were like, what? Let's try that. That would be very convenient for our proteins to do that. And that's because, this amyloid-like, the amyloid formation, this fiber formation, is seen as the last step of a transition between states that your protein can be organized into. So, and the liquid droplet is one of them. Okay, so we would probably be detecting, that was our idea at the time, this final stage there of organization of our protein forming these fibers, these long fibers, right? So we searched for conditions that we could detect the formation of liquid droplets in vitro from using our GRAS proteins. And we were able to see that the one of them, GI81, was actually forming this liquid droplet. So you see there, this is just like a optical microscopy data, okay? So you can see that in this condition, 7.4 of pH and 8% PEG. This PEG here is just an agent that acts as a crowding agent, okay? So that's what it is doing. It's increasing the concentration of molecule in solution. So we can form these droplets there. You can see here with the protein labeled with fluorescent label that the protein is indeed located inside the droplet. We also ran an experiment to show that that's a liquid droplet and not a solid droplet. We have that data too, okay? We showed, that this can happen in several values of pH. That's interesting because then pH can regulate the formation or the disruption of these structures. And we believe, that's our hypothesis, that's what we are trying to do now. It's like from the molecular biophysics in vitro experiment, try to connect with groups in cell biology and test whether we can detect the formation of these droplets within the cell, right? And our hypothesis will be that this membrane-less organelle or this condensate will be the carrier used for secretion of our molecules outside the cell. Keep in mind that if it's membrane-less, there is one last thing you have to do, which is make the molecule to be secreted to cross a membrane. So it doesn't have to cross a membrane because there's no membrane, all right? So it would be much easier for the cell to use these things as carriers for unconventional secretion. We haven't been there yet, or we're not there yet, okay? So we are trying to connect with the cell biologists, and it's tough, right? I mean, it's amazing that, how huge a gap there is between molecular biophysics and cell biology. So we are trying to overcome that with colleagues at the med school in Ribeirão Preto. All right, so if you want to know a little bit more about our GRAS proteins, we published this review years ago, summarizing their biophysical properties. There are lots of questions there, so if we can prove that this condensate is indeed the carrier used by the cell, this explains several things, but opens a lot of new questions. As for, let's say, for example, what defines selectivity? So how the cell knows what goes inside the droplet, how the cell knows what does not go inside the droplet. So we don't know. So if you're going to ask that, don't ask, because I would say, I don't know, okay? So, the take-home message actually is a more general type of message. So this is a moment this is a flourishing field, all right? So there's lots of room for physics, and when I say physics there, it can be as general as possible, experimentalists, theoreticians. Okay, so just to give you an idea, the thermodynamics used to describe the formation of these condensates nowadays is the thermodynamics developed in the 40s and the 50s by Florian Huygens for the homopolymers. So there's lots of room for theoreticians to do some theories on this kind of system. All right? Lots of room for biologists, of course, math, chemistry, and computation. You can even go to applications of condensates. I don't have time to talk about applications. We are trying to do some biotech applications in the group as well, right, to build condensates to perform a specific chemical reaction with a high concentration of an enzyme inside the droplet. So I don't have time to talk about it. So I mean, this is a very exciting moment, you know, for those of you who want to delve into this kind of thing. So there's lots of room, lots of unanswered questions there. As just Nelson said, you know, told us about chromatin. This is also the very effervescent time to be there, right? So I finished thanking our collaborators, the ones who give us money so that we can keep going, keep doing our research in Ribeirão Preto. And of course, thanking you for your attention, and the group who is there hopefully, no, but today is Sunday, so they're not working, I think, okay? So thank you so much.
Thank you. Questions?
These phase separation processes are very interesting. So I'm curious, basically, as you put gas, this protein, you're looking in vitro. Does these condensates have any similarity to what you observe in vivo versus in vitro? And do you know what's inside of it?
Well, in our case, it's just the protein and the PEG, because that's what we put there.
And that's all you have there?
Yes, but usually what happens when we compare condensates from in vivo and in vitro is that the in vitro ones are larger, okay? And of course, you have more control. So
have you tried to change what the?
We've done it all of it. We changed the pH, we changed protein concentration. There is of course a threshold concentration.
But you have protein and what? What's on your solution? Just protein and?
Just buffer. Yeah. Well, salt is a concentration of salt. 3 is HCl. You know, adjusted pH to 7.4, 6, or whatever, and then you push the PEG. So if you don't put the PEG, nothing happens.
So have you tried to put different polymers there, different size, different bigger?
You know what we did? We put the one of the ligands, one of the molecules that have been shown in cell to be secreted by grasp. So we did that. So let's see if the two proteins, the one that's forming the condensate and the one that's supposed to be secreted, if they can co-localize within the droplet, and they do.
So if you change these conditions, can you go from homogeneous or heterogeneous condensate? So they're always homogeneous when you're
ours is homogeneous because it's just one protein.
But
it could be
a protein
heterotypic.
aggregate alone with back with separate.
Yes, that can happen. Usually the most frequent thing that you can find in the cell, at least based on the reports, on the articles, and the papers, is the condensates, the ones detected in vivo. They involve RNA and proteins. So usually
that's right. They have RNA. That's what I said. And so that's why I say you
we
put some larger group and see if this thing happens.
Look, we haven't tested RNA here because RNA would be way out of what is expected for the function of grasps. So
right, but normally when you put RNA, charge play a big role.
We could do that.
So your
we haven't done that.
Condensate's not charge driven.
Yes, we haven't done that. Yeah, that's a good idea. Sure.
Great talk. So is the protein, the IDP, necessary ingredient? Could you get this liquid phase separation out of different groups, of different mixture of globular proteins? What do
you mean, the same? Well, you know what we do? I mean, two things.
Because you said that.
Well, two pieces of information so that maybe will answer your question. So we have, I just told you that the protein has 3 domains, right? 2 PZs and 1 SPR. The SPR is completely disordered. So one thing we are doing that's happening, not right now, but probably tomorrow, the next weeks, we chop it off the SPR domain. And we'll check whether the more ordered domain could undergo phase separation by itself. So I don't have the answer for that. But we have that construction in the lab. So we chop it off. And we did, one thing we did also in the lab in this biotech application was to get an enzyme, a globular one, well-behaved, not expected to undergo phase separation, and we just modified it, adding 2 IDP sequences to its N and C terminus. And then it undergoes phase separation beautifully. So we can control that. You can tune the phase separation using intrinsically disordered sequences added to the protein. And you can also tune you with chart and with other molecules. So we have this Frankenstein in the lab. Why have we done it? I don't know. Right? Just for fun, probably.
Any other question? Okay, thank you again.
Thank you.
So now we have our last lecture by Bill Bialek.
So we'll find out whether the slides worked. So we've been oscillating in scales, and right, it's about it's an afternoon about complex systems, so I guess it's okay. So this will be one last oscillation. The scale will be larger. We're going to talk about animal behavior, hopefully. Very good, thank you. Good, so I'm gonna talk about animal behavior, and let's see. So what I'm gonna tell you about is largely drawn, the core of it is in a recent paper with my experimental colleague Josh Chavitz. It builds on work done earlier. Gordon was a postdoc with us and is now a faculty member at Emory. Vasil was a PhD student and is now a postdoc at Northwestern. Let me point out, when you think about animal behavior, and remember that we're animals, one of the things that's very striking is that it occurs on many different scales, many different time scales. So let's think about a single animal, right? There's also many beautiful things about collective animal behavior. Let's think about the behavior of a single animal. So a familiar one, if arguably one of the most sort of challenging, is to think about the behavior that I'm engaged in now. You know that when we speak, there's structure on the time scale of phonemes, which if we write, they're letters. These come together to form syllables, words, phrases, sentences, paragraphs. And if you think about it, the time scale down here when you're speaking is tens of milliseconds. This can be minutes. If you think about an actor standing up and delivering a soliloquy, you know, you're spanning many, many orders of magnitude, and there's structure on all scales. So one view, which of course comes up in a lot of different contexts in biology, is if you see things happening on different scales, they really are different scales. And for each scale, you should look to a different mechanism. Of course, we know there's another possibility, which is that the systems that generate these behaviors have many, many internal degrees of freedom. When you take a system with a large number of degrees of freedom, there are emergent timescales, which can span many orders of magnitude. And you might even start to think, when you see a very wide range of timescales, you might even start to think about things like scale invariance, where all the timescales get represented more, in some sense, with equal weight. And so these are, you know, kind of the default physicist model and the default biologist model, maybe. I don't know. I don't want to cartoon the sociology too much. So what we're going to do is we're going to plunge, it's a short talk, I'm going to plunge into one example, and we're excited because of what we saw. I'm a theorist, but this is an entirely phenomenological talk. There's a theory, perhaps, there's a class of theories in the back of our mind as we're doing this, but we don't have a theory of the thing that I'm going to show you. And the example that I'm going to show you is drawn from looking at the behavior of flies, not while they fly, but while they walk. Let me make an aside. If you look at, throughout the 20th century, there were two very prominent approaches to animal behavior. One, which is actually grounded in work of physicists in the 19th century, is a subject which came to be called psychophysics, the merger of psychology and physics. And the way that that is done is that you put people or animals in a situation where they're they're looking at something or hearing something or feeling something, and you ask them to make discrete choices among a limited set of alternatives. And you ask how their choices vary as a function of the parameters of the signal, and in particular, you ask about how reliable their choices are. So this, for example, is how we know that our visual system can count single photons. It's how we know that when we're localizing a sound, it's based on the time difference between our two ears and so on. Okay, so there's a lot of beautiful things we said, but the way they succeeded was by simplifying the tasks. You know, did the sound come from the left or the right? That's all you're asking. And the behavior was to make that discrete choice. The other approach were the ethologists. And they went out into the wild and described the behavior of animals in situations that are relevant for their evolution. The sort of high point of this is the discovery of the dance language of the bees, right, which I hope you all know about. It's a beautiful subject. So the descendants of the physicists, they made progress on characterizing human and animal behavior by being very quantitative, but by simplifying the behaviors. The ethologists made progress by sort of appreciating the complexity of behavior in the natural setting, but their description is qualitative. So the question, which started to crystallize in many people's minds around the turn of this century, was, can we allow animals to behave in a more natural way and still be as quantitative as we are in the physics tradition? And so there is now a subject called the physics of behavior. If you go to the American Physical Society meeting next month, you'll find 5 sessions about this. It kind of took off, okay? So here's an example in this spirit. You're watching a fly walk around, and you watch him at very high resolution. So you can see where every leg is, the angles of the wings, how his head is arranged, and everything else. You then take all the discrete images, and you align them, and you discover that the postures, right, the actual shape of the fly as seen in projection, lives in a space. So you have thousands and thousands of pixels to describe the image, but of course the dimensionality of variation is much lower. You project into that lower dimensional space, you look at the trajectories, you discover that the trajectories also have temporal patterns that you can see, for example, by looking at spectrograms, right, because they engage in rhythmic movements and so on. And then you embed these in some way nonlinearly, and what I'm describing is the first thing that we tried now 10 years, now a decade ago, which somehow became ensconced. Probably that was a mistake, but there we are. And in this lower dimensional space, you discover that the probability distribution has many, many peaks. And in fact, the dynamics consists of sitting near one peak, jiggling around a little bit, and then jumping to another one, and so on. And so it makes sense to think of these as being discrete states. And those discrete states come in categories. They correspond to things that people have identified. You know, flies take their wing and they groom their head, right? And so one of these little boxes, an anterior movement, is one of those things. But there's 100 of these states, so they don't all have names. And they're very robust. And for example, if you take different species of flies, you discover the same states. But if you ask about the probability distributions over those states, if you measure the distance between the probability distributions, they're called like Leibler divergence. So this is, yes, the coordinates have lost their meaning. So you do a linear projection of the images, that's not so bad. You then compute spectrograms of the trajectories along each of those coordinates, and then you do a nonlinear embedding. And at that point, the coordinates don't mean very much, but it is important that the dynamics mean something in that you do tend to do the thing that corresponds to having discrete states. There's a discussion here, which I think you're right to be skeptical about this, but this is going to be a starting point. So you have these discrete states. If you look at different species of flies, the distributions are similar, and the degree of similarity recapitulates the evolutionary tree, which we found kind of surprising, actually. Actually, that was Gordon. We worked together to identify all this, but it was Gordon who figured that out about different species of flies. Okay, so how do you characterize, so this says that behavior is a sequence of states. So let's say fly alpha is in state I at time t. There's a variable si alpha equals 1, and otherwise it's 0. So it's sort of Potts-like. You say that there are correlations that extend in time, so you say, let's compute a correlation function. Too many indices, right? So who cares which fly it was? Let's average over flies. And actually, for the purposes of this discussion, do I care whether it's correlated? Is he coming back to the state where he grooms his right eye with his right front leg or his left eye with his left? I don't care about that either, so let me also average over all the states. And so this is basically the probability. The way you measure correlations is just ask, if he's doing something at time t, what's the probability that he comes back and does the same thing at time t prime? Good, so you compute a correlation function. Unfortunately, this includes a disconnected part. So in order to make a meaningful correlation function, I should subtract the disconnected part. That is to say, I should subtract the means. But how do I compute the mean? Well, one way I can do it is to say, well, look, I record for an hour. And the correlations are presumably on the time scales of seconds. So I could take the mean of every variable to be its average over the hour. Unfortunately, if you do that, even if you have a very well-defined correlation time, if the correlation time is several seconds, you distort the correlation function enormously. So then you say, okay, well, that was a bad idea. Let me try instead doing the experiment on many different flies, and let me take the average to be the average over flies. Well, that's a good idea, but if 1% of the variance, 1% of the behavioral variance is actually individual, then you distort the correlation function the other way. And so even and you all know this, but normally when we do physics, there's no problem, right? Because if I compute the spin-spin correlation function, even if I get to a correlation length of microns, you know, my sample was millimeters, it's fine. You don't have to worry. So this is a problem if you're trying to uncover a single exponentially decaying correlation. This is a log scale. It's even worse if the underlying correlations are truly scale invariant, because of course they always have a short time cutoff, and then these different strategies for subtracting the mean distort the long time behavior, and you just make a mess out of things. This problem also comes up, by the way, in analyzing collective animal behaviors. And there are ideas that are not unrelated to what I'm going to tell you about. But anyhow, okay, so this is a problem that finding these long-time correlations is very hard. There's various mistakes you can make. And so what we decided to do was to say, let's do something that in some sense goes headlong into the mistake you could make. So let's look at a window of size capital T and estimate the probability that the system occupies that an individual alpha occupies state I as an average over that window. Now, if the only thing going on is that there are statistical variations, then you know that this will be a random number, but its variance will go down as 1 over t, right? The usual. So let's compute that variance, and in fact, let's compute the variance across individuals, and again, let's get rid of which state it is, because I don't care. So if there's short-range correlations, your statistical intuition is correct, the variance should fall as 1 over t. If there are individual differences, it will plateau at the value of the variance among individuals. But interestingly, if there are long-range or scale invariant correlations, right, then you should see a non-trivial power here. And that's where it should show up. And so this is also a little bit like, right, we're computing an integral. We're looking for the correlations in S by computing something which is an integral over time. I'll remind you, this is just like when you think about anomalous diffusion, right? The interesting correlation function is the correlation function of velocities, but it's easier to measure the displacement, so you measure the integral. Same idea. Good. Okay, so let's try it. There's a data set with 59 flies, each of which were recorded for an hour. The frame rate is 100 hertz. We go through this whole analysis of defining 100 discrete states, and then you compute the correlation function that this phi 2 that we defined, and you see this. And you think, well, boring. It decays rapidly and is plateauing, and that's because there are large individual differences. But that's because you only looked out to 1 second. So let's look out to 100 to about a minute or so, a couple of minutes. Now it's useful to have a logarithmic scale on the y-axis, and now maybe you're not so sure where the plateau was, but then if you actually look over the full range of the data, you see that there's structure continuing out as far as we can go, out to an hour. And in fact, from about 7 seconds onward, it's an extraordinarily good fit to this anomalous decay. And the predicted variance in the background is this. Remember I told you that individual differences can look like long-range correlations, essentially because you remember your identity forever. But it's also true that if there are long-range correlations, you overestimate the individual differences because it's not enough time for the individuals to be ergodic, right? So you have too much memory. So if this is correct, then of course we can subtract an estimate of the background. In order to do the things that are going to happen on the next slide, you have to be extremely careful about error bars. And so let me just say that, and if somebody's curious, we can talk about it. If you subtract the background, you see a clean power law over 3 decades, which is essentially the maximum that you could possibly see in these data. Although the frame...
That is not real, right?
Sorry?
It's not real. It appears that it makes
this
change concavity here.
Well, there's a bend on this side, and that's because that's below 1 second. And although the frame rate is 100 hertz, the thing we call a state includes things like rhythmic behaviors, right? So in order to identify those, you need a window in time. And that window actually reaches out to about here. So the scale invariance kicks in as soon as you're past the window for defining states. And so that means that you essentially, and it extends out to the full length of the data, so you see scale invariance over essentially the entire span that's possible in the experiment. And you'll notice that when you fit, you get exponents with very small error bars because this is not approximately a power law, right? Or rather, it is a very good approximation to being a power law. But remember that when you think about measuring, if you think back to critical phenomena, right? You can measure many things if there's some limited number of underlying primary scaling fields, then many correlation functions that you measure should show you the same exponent, possibly with different coefficients. So for instance, instead of looking at the second moment, you could look at a third moment, and then you should see a 3 delta appear somewhere. And we define state by looking in each frame of our video, but I could put 2 successive frames together, and then instead of having 100 states, in principle I have 10,000, but you only visit about 1,000 of them. So you do the same thing, but with a time resolution that's 1/2. But unlike the usual thing where it's just the average, right, where you coarse grain, here it's more complicated because the states combine in a non-trivial way. So if you do this, the 2 successive state thing, of course, gives you back the same exponent. And the 3rd moment gives you something which is steeper, but by a 3 instead of a 2. So, what have we done? Strangely, we have 2 copies of the same slide. Okay. So what I've shown you is that we can see scale invariance over 3 decades, which is the maximum possible in the data. The exponents are reproducible in the 3rd decimal place. And there are different correlation functions you can measure which are all consistent with the same underlying scaling field. That's what we know. So the dream, of course, is that the reason this happens is that the neural network that's generating this behavior has somehow organized itself to some scale invariant state. And it is that scaling that we're seeing reflected in the behavior. And it's late, so I'm not going to go to the blackboard and tell you anything, but let me just make an observation. If I tell you that you have a correlation function that decays as a power law, you all know that you can rewrite that as a sum over many, many exponentials, as long as you get the distribution of rates right. Okay, you all know that. And that could be for an interesting reason, that those are in some sense the normal modes of this interacting system, or it could be an uninteresting reason, which is that you just happen to have an inhomogeneous system and you're averaging. So some of you will know that this was an argument in the discussion of 1/f noise way back, right? So in certain classes of materials, 1/f noise is the uninteresting thing. It's electrons getting trapped and you have a distribution of trap depths which generates a long distribution of timescales, and so it looks like a power law, but it's not. The alternative, as I say, the alternative is that you really have, these are the modes of some interacting system. You should think about what happens if you try to do this decomposition and you want to predict the 3-point function instead of the 2-point function. That's a lot harder. So if I just give you weights to the different timescales, so the 2-point function comes out right, and you compute the 3-point function, I don't think you're guaranteed to get scaling with 3 delta instead of 2 delta. You really need something non-trivial, like in our understanding of real scaling systems. So I think that we're seeing signs of scale invariant behavior that are surprisingly precise. I want to remind you, these are flies walking around in a featureless environment, and yet you see these very reproducible things. Okay. And so we're excited. The next generation of experiments will look at many more flies over much longer timescales. And actually, if you think about it, the range of timescales and the precision that we'll be getting to is actually comparable to the precision with which scaling is tested in critical phenomena. So we're getting to the point where you maybe will see phenomenology of a biological system which is as precise as fundamental physical things. So let me stop there.
Thank you.
So let's just be a little provocative just for the fun of it. Isn't scale invariant boring? Why behavior should be there? There's no information into scale invariance. So why the behavior should fall on that and shouldn't break this if there is some information into the process?
So we're looking at a behavior which is, if you will, spontaneous. The animal's wandering around in a featureless environment. So you could argue that what you're probing are the internal timescales that the system has accessible to it. If it finds itself in an environment in which something's happening on some timescale, it is going to lock into that and do something that's appropriate to that timescale.
Is that what you're going to observing? Have you had the experiment to show?
I think that's what most people observe in most experiments.
No, but it would be nice to see if you can do this transition. I would like to see the transition from one to the other. Is that going to be just a very sharp transition, or is that something that you are going to perturb from scale invariant to?
So, we don't know. We haven't.
That's pretty cool.
Yeah. So as I say, I think this is, you're observing the sort of ingredients of behavior, right? By taking away all the external things.
More questions?
Now let me counter that and say that the scale invariance, or this non-exponential decay, is a very interesting phenomenon, because it says that there must be multiple time scales in your system. So as you said, if you compound your correlation function, assuming that some parameter is fixed for the, at least it has a lower, it's lower dynamics, then you average that over some distribution, you are bound to get some either power law or some non-exponential decay. So you indicate that that might not be the case, but I was wondering if that's what you're not seeing is multiple time scales in your correlation function, so that when you average them over, you get this power law-like behavior.
So I think, okay, so the fact, let's see. Let me see if I put an alpha here. What do I want to do? I want this to be k to the alpha minus 1, I think, if I can do the algebra in my head. That's an identity, right? So there's no argument that seeing a power law is the same as seeing a superposition of timescales. The question is, in order for this to really work, this statement has to be precise, has to reach a certain level of precision across a certain dynamic range. So one question you could ask is, How precise is that? And at some point, do you say, so for instance, I can make those correlation functions with a superposition of 6 timescales, and you won't be able to tell the difference. Unfortunately, you have to be sure that the amplitude associated with each timescale is proportional exactly to a power of the timescale. So it's not a generic explanation. You've just transferred it to somewhere else. The other question is, if I tell you that this thing goes like t to the 2 delta, that can be gotten from something like this. But then that's not guaranteed, right? You need to now exactly how you transform from the sort of the modes that generate the individual timescales to the variable that you observe. That gets more complicated. So I don't know. I mean, I told you it's a phenomenological talk. I would also say, partly in answer to both questions, the literature about scale invariance and critical phenomena in biological networks has a lot of volume associated with why it should be true, and rather less volume associated with whether it is true or not. So we've tried to stay away from that.
So the fact that the flies will eventually get tired should change this, right?
So it's very interesting. Yes, I mean, well, eventually they die, actually, which, for flies, it's not forever. So, well, not for us either, but the time scale, it's not forever on this time scale. So, for instance, you can do an experiment on flies over a week. You have to figure out how you're going to feed them. You want to be sure you don't feed them in a rhythmic way because that's going to produce things. But you, of course, do turn the lights on and off. And so anything you see is going to have the circadian rhythm. I can look for 1 hour and you say, well, why did you stop at an hour? Well, if you go to 10 hours, you're going to start to see the curvature, right? And 10 is almost 12, which is half a day. And so you're going to see something which starts to betray the circadian rhythm. So one of the things we're spending time thinking about is how do you disentangle these things? And actually, so the simple thing you can do is to take chunks for an hour at different parts of the day, and it looks like even maybe in some parts of the day you don't see scale invariance. So we'll see.
Maybe you have already answered, but I'm just curious because this means, this kind of power law, that there are no scale in the fly, but it has a size, it has a time of life, it has a biological cycle.
Between a second and an hour, there's no characteristic scale. Between 1 second and 1 hour, maybe that's what it's called.
But what that determines that?
No. It's not that there isn't a time scale. It's that there are all time scales, right? Oh, sorry. There's this, is scale invariance the statement that there is no scale or that all scales are represented? It depends it says there's no single characteristic time scale. But the thing is that when people study a behavior that seems to happen on one time scale, they talk about the time scale and where it comes from. But then, for example, you think about learning, there's some time scale. But if you look at it, or forgetting, but if you look at a synapse, which is supposed to be the site of some of this learning, there's a network of 100 proteins that interact to determine the strength of a synapse. Do you think that the dynamics of that network is characterized by a single time scale? That would be a big surprise, right? So who knows, right? Maybe there's, yes.
Okay.
Just one quick question. How universal do you expect this to be? For instance, imagine that you take pictures of ants or any other animal. Do you expect to find it in the same kind of behavior?
So I'll give you the answer that we wrote in a paper where we saw scaling and the dynamics of networks of neurons. That, you know, before you ask about universality, you should ask about reproducibility. So about reproducibility. So if you do the same thing again, do you get the same answer? Which, you know, when you're talking about animal behavior, is something of a triumph. So we think we've gotten that far. You know, we'll see.
Any more questions? No, unless.
Just getting back to the very beginning of your talk, when you talk about the discrete states. So couldn't you somehow eventually saying that, well, this invariance has some limits in terms of associated to the behavior, but as you said, well, one time it suddenly it dies and then breaks your story.
Okay, so aside from obvious, you know, there's some obvious cutoff at the two ends.
Exactly.
But the question is in between. So even if I give you the example in language, right? Phonemes vary in duration by a factor of 3. Syllables vary in duration by a factor of 4, and so on. So every one of these discrete timescales actually has a range. And if the ranges start to overlap, then the idea that there were discrete timescales maybe isn't so useful anymore.
Okay.
Okay, thank you again. I think there's some pizza upstairs. Or there will be pizza.